All right, so very good morning to all of you, and welcome to AP India webinar series, webinar number 12. And today is a very interesting session we have. So would like to welcome officially to all of you. And I think we will just get started in a minute from now. So once again, warm welcome to all of you. Just keep your lines muted and just wait for another 1 minute to get started. Thank you.
So very good morning and good evening for all the participants and speakers. Today we have at webinar 12 under CMC section for APA India 2021. Under CMC, which is Chemistry, Manufacturing and Control, on today's week, we are going to discuss more about the excipients, stability issues, impurities, extractable and leachables, and drug excipient compatibility studies. So as a first speaker, we have Professor Suri, and he is going to talk on excipient phase behavior in frozen and frozen dried systems, which is a potential implications in drug stability. After that, I'm going to talk on drug excipient compatibility studies, quo vadis, which is like where we are going on. And then from Baxter, Monish Shahai will talk on extractable and leachable studies for complex LVP drug products, which is a technical challenges and considerations. Followed to that, Amjad will talk on organic impurities in pharmaceutical industries and USP perspective. So on today's webinar, we all are going to learn few things on drug product development, where the impurities, implications of impurities, excipients, drug interactions in injectable or lyo products, how the excipients role, how their phase behaviors are impacting on stability, and so on. So with this quick introductions on today's topics, I would request Dr. Anurag to introduce Professor Suri and kickstart our presentation.
Thank you, Ravi. So this was Dr. Ravi Shah. He's the chair. He's the moderator and the chair for this today's session. And Dr. Anurag is going to be the moderator. So thank you, Ravi, for your introductory remarks and kickstarting the session for today. So since we have some more time and participants are still joining in, so very quickly, Dr. Anurag, if it's okay, we can just go through a few slides on APA so that meanwhile more participants will join in, and then we can introduce our first speaker. Is that okay, Dr. Anurag?
Absolutely,
Sure.
yes.
So once again, thank you, Dr. Ravi, for your remarks, introductory remarks, and let's get started with that. So this is webinar number 12. Already, Dr. Ravi has explained in detail the session, how it's going to take place, and some background about today's session. So we would very quickly like to thank all our organizers, including the presiding chair and chair-elect of each of the committees, because it is their contribution, their active involvement, because of which we have such an interesting agenda on board. So a big thank you to the presiding chair and chair-elect of all the committees. Special thanks to all the committee members, because chair-elect and president, the chair, they are supported by a very active committee. So big thank you to all the committee members in regulated bioanalysis, in discovery bioanalysis, biotransformation, and CMC. So without the support from sponsors, again, it's not possible to conduct such an international meeting consistently for, I think this is our 12th meeting. So a big thank you to all our sponsors, and would like to mention that most of the sponsors have been with APA for more than a decade now. So a big thank you to all the sponsors for their support. Shimadzu, SciEx, Zepsci, Phenomenix, Thermo Fisher, Agilent, Syngene, USP, NATCO, Vida Clinical Research, Bristol Myers Squibb, Zoetis, Nanotemper, In Vitro Edmet Labs, Origin Discoveries Technology Limited, Wuxi Aptech, and Loris Infosystems. So big thank you to all our sponsors. We would also like to thank our collaborators, Life Science Sector Skill Development Council, which comes under Ministry of Skill Development and Entrepreneurship. So they are our collaborator this year, and we would like to thank them as well. Very quickly about the poster session, big thank you to all our participants, because we received more than 45 posters for the poster session, which is going to take place on 2nd of October, which is next Saturday. So what we did is, like, since there were many posters, so we had to shortlist the posters based on how they're aligned to the theme and to the different areas which AP India meeting covers. So based on that, we have shortlisted the posters, and all the shortlisted poster presenters they would have received an email with all the details, like what next steps needs to be taken. And I think most of them have already registered also now with a special discount code which was shared with them. And the final poster session will take place on 2nd of October. So for them, I would like to request to get started with their preparations for poster presentation. Since it's going to be in a virtual mode, so each one of you will have an opportunity to present your poster. You'll have 8 minutes, so 6 minutes for your presentation and 2 minutes for Q&A from the jury. So good luck to all the shortlisted poster presenters. Just to remind you, in total, there are 9 prizes, very interesting. I mean, there are cash rewards, there are trophy and certificate and other interesting prizes attached to it. So there will be 3 first prize, 3 second prize, and 3 best prize. So I would like to wish all the shortlisted poster presenters good luck. And very soon you'll receive further instructions for the next Saturday, 2nd October poster session. Again, as usual, we always do a Kindle draw at the end of the webinar. So in today's webinar also, once all the talks are over, we'll be doing a Kindle draw. So would like to request all of you to participate in that. So to participate in that, you need to submit your response when we make the poll online. So at the end of webinar, when all the 4 talks are over, we will display the poll, the survey, and you need to submit your response, and then you will be eligible to participate in the Kindle draw, which will take place at the end. So, so far 11 webinars we have done, and 2 lucky winners we have selected from that draw. So around 22 winners we already have with us. So congratulations to all of them. Quickly, for all the participants who are joining for the first time this webinar series, as a participant, your videos and audio would be off. So if you have any question, queries, or for the speaker, feel free to use the Q&A tab, or in a chat box, you can post your questions. And after the talk is over, the moderator will take up your questions and address and will take up those questions with the speaker. In case during the webinar, if you are not able to, if your query is not addressed, then feel free to post your queries on bostondb21@gmail.com. We'll reach out to the concerned speaker and get your query clarified. Again, to participate in the poll, you need to, do participate in the poll at the end of the webinar to be eligible for Kindle draw. And finally, once we are on the last month of the webinar, which is in November, we intend to upload all these talks, recorded versions on our YouTube channel. Of course, we'll seek permission from each of the speakers, and all those speakers who gave their consent, those talks would be published on our online, YouTube channel of Boston International. So we'll keep you posted on that as well. So I think with that, I think we can get started. Once again, a big thank you to Dr. Ravi for his introductory remarks. Would now like to invite our moderator, Dr. Anurag, to take the proceedings further and introduce our first speaker. Thank you.
Excellent. Okay. Good morning, good evening, everyone. Thank you for joining on a Saturday morning, evening time, Friday evening for Dr. Suri, right? So it's my pleasure to introduce the speaker of the day, the first speaker of the day, and there cannot be better way to start this theme other than having Dr. Raj Suryanarayanan as the first speaker. And Dr. Raj Suryanarayanan, very popularly known as Suri, is a very familiar name in the pharmaceutical industry and the academia world globally. He is professor and William and Mildred Peters Endowment Chair in College of Pharmacy, University of Minnesota. He has his roots back here in India with his bachelor's and master's degrees obtained from PHU. Then he continued his educational journey, earning higher degrees, MSc and PhD in pharmaceutics from University of British Columbia, Canada. He's been very actively researching and presenter in the area of principles of pharmaceutical material sciences to design robust pharmaceutical dosage forms with reproducible and predictable properties. His team, research team, has developed and innovated in this area several innovations to his credit. And they've developed, for example, low temperature powder XRD diffractometry techniques to study frozen and freeze-dried pharmaceutical systems, very unique in itself. He's a consultant to numerous pharmaceutical companies. He was a member of USP Expert Committee on excipients test methods. He is very active in several other professional activities. He's member of apps. He is past chair of teachers of pharmaceutical selection of American Association of College of Pharmacy. He is a member of Academy of Distinguished Teachers at University of Minnesota, recipient of several awards, amongst which APPS Outstanding Educator Award, David Grant Research Achievement Award in Physical Pharmacy, and PHRMA Foundation Award are some of key accomplishments. He is
Anurag, your voice is little breaking. Yeah.
Of molecular
Anurag, are you there? I think there's some connectivity issue at his end. So I think he already introduced our first speaker, Raj Suryanarayan. So I think where we lost him was on the last few sentences where he was talking about the achievements of Dr. Raj. So as he already mentioned that Suri is actually a member of Academy of Distinguished Teachers at University of Minnesota. He's also the recipient from APPS of the Outstanding Educator Award and the David Grant Research Achievement Award in the Physical Pharmacy, and the Pharmaceutical Research and Manufacturer of America Foundation Award. And he's an associate editor of Molecular Pharmaceutics. So we are so very honored to have such a distinguished, esteemed speaker with us, Raj Suryanarayan from University of Minnesota. So without further ado, would like to request Raj to stage is all yours, and please get started with your presentation. Thank you. Looking forward to your talk, Raj.
Thank you so much. Thank you, Anurag, for the lovely introduction. Let me see how I get my talk in. Okay. Can you see my screen?
No, not yet. I think you'll have to share your screen. Yeah.
Okay, 1 second. Screen. Can you see my screen now?
Yes, and you had to go to the first slide. Yeah, great. You're all set, Raj. Thank you.
Thank you so much. Thank you again for the invitation and for this opportunity to speak to this group. So today's talk, I'm going to talk about some of the excipient phase behavior in frozen and freeze-dried systems and what implications these can have on drug stability. So by way of acknowledgement, we got funding from a number of sources which are listed here, and the students and postdocs who are involved in the work will be listed as I go along in the presentation. So for those of you who are not very familiar with the process of freeze drying, I'll just provide a brief introduction. In freeze drying, what we do is that we take a solution which we are going to freeze dry. So because we have not yet done the freeze drying or lyophilization, we refer to it as the pre-lyo solution. And when we freeze dry it, this is the type of cake that we want. We want an elegant cake where the powder forms a nice mass that is able to hold its own weight. During the freeze drying process, if the solutes do not crystallize and they remain amorphous, then they are characterized by something called the glass transition temperature or the freeze concentrate. And during the freeze drying, if you go above the glass transition temperature, the cake will collapse and you get a cake that looks like this. As you can see, this is not an elegant cake. On the other hand, if the solutes were to crystallize and then the system is characterized by a eutectic temperature, and if the practitioner of freeze drying is not careful and freeze dries above the eutectic temperature, we get what is called as the melt back. And for anybody who is doing freeze drying, both cake collapse and melt back are not desired. These are things that you want to avoid. So in order to develop a robust freeze drying cycle, we often try to monitor the transitions during the various stages of the freeze drying process. This can be a challenge, but it's very valuable because it gives us very valuable information. And for this purpose, often we will use multiple analytical techniques. We will just focus on one or two today, but for those of you who are interested, you can see in the literature that there are many techniques available. The most important message that I want to leave you with is that when we freeze dry materials, we have to use excipients judiciously. More is not better. We need to optimize the concentration and use only what is needed. And this is doubly important because a lot of the freeze-dried products are intended for parenteral administration. And when you're giving it by the parenteral route, you obviously want to keep your excipient concentration at the minimum possible. The other thing that I hope to show you today is that these freeze-dried formulations are complex and there is potential for interaction between the formulation components. And how do we exploit these interactions to make a robust product? That's what we are going to look at. Now, for those of you who are in the area of material science, this slide would be something that you probably have seen in your graduate work, but for others, I'll walk you through that. Now, for every formulation component that I have in a freeze-dried product, the solid state of the material is going to be function specific. What do I mean? Suppose you are freeze drying a small molecule such as an antibiotic or an anti-cancer drug. If you get the drug in the crystalline state, you're going to get a long stability. Crystalline materials are more stable than their amorphous counterparts. But the price that you're going to pay is that the crystalline materials will dissolve at a slower rate, so it may translate to a longer reconstitution time. So if I have a small molecule that is thermally unstable, we will make it crystalline and live with a longer recon time. On the other hand, if I have a small molecule that is very, very stable, getting it amorphous would give us a value of rapid reconstitution. When we come to proteins, most of them are difficult to crystallize, so I'm not going to talk about them. Bulking agents are materials that are added so that we get an elegant cake. If you recall, we get a very nice looking cake here because we often have added a bulking agent such as mannitol or glycine. And these bulking agents, we want them in the crystalline state to exert their function. On the other hand, when you look at buffers, buffers are often a combination of a free acid and its salt. So if there is selective crystallization of a buffer component, if one of the components crystallizes, there'll be a pH shift, and that can be hurtful to the formulation. So with buffers, we want them to be amorphous. And finally, when we are freeze-drying proteins, the stress of the freezing and drying can denature proteins. So in order to prevent that, we often add materials called lyoprotectants. Disaccharides such as sucrose and trehalose are two examples. And for these to exert their action, they have to be amorphous because they have to interact with the protein, which means they cannot phase separate. So they have to be a part of the amorphous phase. So as you can see here on the right side, every ingredient, be it the active or the excipients, we have very specific physical form requirement based on the function. Now, if we take phosphate buffer, for example, which we use to buffer solutions, the phosphate buffer has a long history of use. All the green here tells us the good properties of this buffer. It has a high buffer capacity at pH of interest, which means I can use a very low buffer concentration. And things such as phosphate buffered saline have been used for a long time. So we have a very good understanding of how these buffer systems behave. However, we also have recognized that there are some problems with this buffer, which limits its use. So which will really be the first case study that I'll talk about today, how there can be a buffer salt crystallization during freezing and what effect that does that have on the protein behavior, specifically the aggregation of the protein. And we will look at this under two contexts. As I change the buffer concentration, which is a formulation part, and as I change the protein concentration, which could be a dose issue in a formulation. And the second case study that we are going to talk about is sugar crystallization. And again, going back, as I mentioned, we want the sugar to remain amorphous. And what will happen if there is sugar crystallization? Can it affect the protein? And in this case study, we will look at it in the context of co-solutes and also proteins, which are the active ingredients. So to give you a flavor of what buffer crystallization is, what I have plotted here is the solubility as a function of temperature for the two buffer components, the monosodium and the disodium hydrogen phosphate. And you can right away see that the disodium hydrogen phosphate has a much sharper temperature dependence, which means the solubility goes down very quickly as you lower the temperature. And so this salt can selectively crystallize out of the system. And when it does that, the pH can go down up to 4 units. And you know that pH is on the log scale. So we are talking about 4 orders of magnitude change in hydrogen ion concentration because of the selective crystallization of the one buffer component. And if this were to happen, as you can imagine, if the protein is stable only over a narrow pH range, we run the risk of destabilization of the protein due to crystallization of the buffer salt. Now, one can question if sodium phosphate gives such a problem, why do we use it? And the answer to that is that we have a very long history of using this buffer. This buffer has a lot of good properties that we talked about. And as a result, the industry continues to want to use this buffer in formulations, not always, but in many cases. So when we lower the temperature, how does the pH change? What I've plotted here is the temperature as a function of time. As we are cooling the buffer, we are looking at two buffers, concentrations 100 millimolar and 10 millimolar. And the black line is the temperature as a function of time because we are cooling it. So we can ignore that for a minute. If you look at the pH, which is the red line, you can see that we are starting at a pH of 7. And then the crystallization of the buffer component causes the pH to go down to 3. As we know very well, this is something that's well documented. If you change the initial buffer concentration, we have a lower buffer concentration, things get better. We go from a pH of 7 to about 4.5, but still there is a pH shift. The message that I would like you to take from this slide is that the concentration of the buffer plays a role in the degree of pH shift. High buffer concentration, larger pH shift, which means potential for more problem. Is that indeed the case? What we have here is the X-ray diffraction pattern at low temperature of sodium phosphate buffer. So we are plotting the intensity as a function of 2 theta. Even if you're not familiar with X-ray diffraction, it's not important. You can, that message I'll convey here. We are cooling the system from 0 to -40. And as ice starts to crystallize, these are all the peaks of ice. Any crystalline phase has the peaks, and that's shown here. But the disodium phosphate that crystallizes, it also shows peaks here. You can see that as the temperature goes down, the peak increases in intensity, meaning that there is more crystallization happening, which is something that we know very well. Now, if you lower the buffer concentration, we still see buffer salt crystallization, but the magnitude of crystallization is less. Not very surprising. We have lower solute concentration, so less is crystallizing. Now, we are going to now look at this system with the protein present in it. So when you have a protein, a large molecule in a frozen state, the protein can inhibit the buffer salt crystallization. So what we have here is phosphate buffered saline, PBS, initial pH 7.3. When you take it down to -25, there is about a 4-unit drop in pH, big drop. If you just look at BSA in PBS, now bovine serum albumin at a concentration of 10 milligram per mil, the pH shift is only 1, which means the protein is able to inhibit the buffer salt crystallization and minimize the pH shift. You take 10 millimolar sodium phosphate, you see a 2.8 pH shift. You put protein, right away, there is attenuation of the pH shift. The system shows much less change in pH. You increase the buffer concentration. We go from a pH shift of 4.1 to 3.1. Not as big a change because now I've got a high buffer concentration. So the buffer is able to overcome the effect of the protein. But the real question that we want to ask is, what is the effect of this pH shift on protein stability? And in this case, the way we are going to measure the protein stability is by measuring the aggregation. So we are plotting the particle count as a function of the different processing. So if you look at the y-axis, you can see a count up to 24,000. So if I have a higher count, that means that more particles are aggregating. So if you have the phosphate buffered saline, in which I'm dissolving albumin, you can see that initial about 3,000-4,000 counts, you go to 5 freeze-thaw cycles, a small change, not a big one. 10 millimolar phosphate, exactly the same. The protein is able to inhibit the buffer crystallization and the protein remains stable. You go to a higher buffer concentration, you can see that there is a dramatic rise in the number of particles. Now we are getting about 20,000 particles after 5 freeze-thaw cycles. And the reason for this is the pH shift. We had already seen that there is a pH shift with a higher buffer concentration, and that pH shift is known to destabilize the protein, resulting in a high particle count due to pH shift. Now, if you were to take the same study in just water, you see that the particle count is not very high. And you had seen with 100 millimolar sodium phosphate how we saw this big increase in particle count. And we are attributing this to pH shift because of buffer salt crystallization. Then the question becomes, if there is some way in which I can prevent the buffer salt crystallization, can I prevent the particle aggregation? And the answer to that is indeed yes. If you use 5% cellobiose, which is a non-crystallizing solute, which is able to prevent the buffer salt crystallization, then you can see that the change in the particle count is much less, like in the other cases. So the take-home message here, as we saw earlier, if you were to have buffers, and if there is crystallization of a buffer component, then you run the risk of a pH shift that can cause destabilization of the protein. So this is a very good example where you need to be very careful with the concentration of your excipient. A lower concentration of excipient is much more effective than a higher concentration. This is something that one would not think about. It won't be readily apparent, but the studies clearly show that. But now I want to move to the second case where we are going to look at the effect of sugar crystallization. So we saw earlier that the lyoprotectants such as sucrose or trehalose, we need to keep them amorphous. So if they crystallize, then we run the risk of the lyoprotectant not able to exert its stabilizing action. And we will look at this under a variety of conditions such as annealing, presence of a co-solute, and the presence of a protein. So here is why we need the lyoprotectant in the amorphous state. The conventional wisdom is that when you had a sugar, what the sugar does is that it hydrogen bonds to the protein. So once it hydrogen bonds to the protein, it is able to prevent the protein from denaturing, and that way the protein remains stable. However, if the lyoprotectant crystallizes and phase separates, its ability to hydrogen bond with the protein will be removed, and so there is the risk of protein destabilization. And people have shown that when trehalose crystallizes, you can start to see protein aggregation. So this is something that has already been shown. So how do we do these studies? We do these studies using high intensity X-rays. I won't go into the details, but to give you the high level view, we got the sample in a cuvette here, and we have got an X-ray source. The X-rays go through the sample, and we have got a two-dimensional detector where we are able to collect the entire Debye ring. And then you can break down this ring and get what looks like a conventional X-ray diffraction pattern. So now, when we took trehalose solution, trehalose is thought to be stable for a very long time. But when we did these studies where we anneal the sample, and annealing is something that we do to facilitate solute crystallization, what we observe is the peaks of hexagonal ice, the green ellipses that you see here, but we also saw several peaks that could be attributed to trehalose dihydrate. In other words, trehalose was crystallizing as a dihydrate in these systems. But this was very surprising because people had done a lot of studies earlier and nobody had seen trehalose crystallization. And so the question comes up, why was trehalose crystallization not reported in the literature? And to answer that question, if you were to do the X-ray diffraction work, what you observe, so I'm going to go from the bottom to the top. What we are going to do is we are going to freeze dry the system. And during the freeze drying, we are obtaining the X-ray diffraction pattern. So we are able to monitor the system as the freeze drying happens. So we have got primary drying at a very low temperature where ice sublimation happens, the ice peaks will start to leave. But even when the ice peaks leave, we are able to see a lot of peaks due to trehalose. So trehalose does crystallize. But then what happens is that as you continue the drying process into the secondary drying and remove the water, the trehalose undergoes a transition from a crystalline state to a very poorly crystalline or amorphous state. Now, this is extremely surprising because a crystalline material is not expected to spontaneously convert to the amorphous form. But the reason this happens here is that in the frozen state, trehalose crystallizes as a dihydrate. And when you dry it, we dehydrate the material, and the dehydration causes lattice collapse and formation of an amorphous product. So we have got two things going on here, dehydration coupled by crystalline to the amorphous transition. And then when you look at the final lyophile, it looks like a broad peak. So people thought that the solute was retained amorphous. That's not the case. It crystallized, phase separates, and then undergoes the crystalline to amorphous transition. Now, when we use trehalose, we often will use it in combination with mannitol. Mannitol is the bulking agent. So we've got 4% trehalose and 2% mannitol. And we are looking at the X-ray diffraction pattern of this mixture as we are annealing it at -18 degrees centigrade. And the key message that I want to convey here is that we first see ice peaks, then we see mannitol crystallization, and then we see trehalose crystallization. Now, I previously told you that I really want mannitol to crystallize because that will give me an elegant cake. But the bad news is that if mannitol crystallizes, it will promote trehalose crystallization. And how well does this happen? If you plot the intensity of the peaks as a function of time, you can see that the mannitol crystallization is happening here, and the trehalose crystallization nicely follows the mannitol crystallization. So a crystallizing solute can promote the crystallization of the second solute. Then the question becomes, what will happen if I were to add a protein? Proteins don't crystallize. What do they do to trehalose crystallization? And the answer is shown here. We have got peptide dehydrogenase, 2 milligrams per mil, trehalose and mannitol. Again, we are annealing at -18. And mannitol crystallizes quite readily. That's not surprising. But in presence of the protein, the trihalo's crystallization needs 44 hours of annealing. That is something that we are not likely to do. So you really have to hold it for a very long time before there will be crystallization. So the message here is that the protein inhibits trihalo's crystallization, and this effect is concentration dependent. Higher the protein concentration, more the crystallization inhibition. So what's the sequence of events? Protein inhibits trihalo's crystallization. Trihalo's is retained amorphous. Amorphous trihalo's functions as an effective lyoprotectant and stabilizes the protein. So you can see here that there is a very nice synergistic effect between the active ingredient and the excipient. The protein kept the lyoprotectant amorphous, and by retaining it amorphous, it was then able to exert its stabilizing action and was able to keep the protein stable. So there is a complex interplay of events in these formulations, and the careful formulator would have to look for these things so that we can make robust formulations. So let me conclude my talk. So when we select excipients, particularly when we are dealing with complex freeze-dried type products, the product stability hinges on excipient functionality. So the physical form of the excipient can be critical, and we need to be very careful with excipient concentration selection. It has to be optimized, and more is not necessarily better. And finally, as a formulator, we want to absolutely minimize the number of components in our formulation. So we really want to find excipients that have multiple functionalities that will help us simplify the formulation. And that's the area in which we are doing our current research, identifying excipients with multiple functionalities. So what we are seeing here is a complex interplay of drug and excipients. The API can influence the excipient behavior. One excipient can influence the behavior of a second excipient, and it'll be good to get a fundamental understanding of these types of interactions. And when we deal with processes like freeze drying, there are numerous processing steps. There is potential for phase transitions, and monitoring them can be a challenge, but they can be very valuable to the formulator because they give information that can help us make formulations that are robust. And often we will need multiple analytical techniques for this purpose. So I will stop here. Once again, I thank you for the opportunity, and I'll be happy to take questions.
Thank you, Raj. Can you, would request you to stop sharing so that we can highlight your video?
Sure.
And would request Anurag to take up the questions further.
Yeah. Well, thank you, Suri, for nice presentation. I can see just one question at this point of time. And the question is, why proteins inhibit the buffer precipitation?
Okay. Yeah, this is almost a universal behavior, and we can attribute this to the fact that proteins are large molecules. They intrinsically resist crystallization. So when we are cooling the system and there is ice crystallization, and then we have got the freeze concentrate, and the protein is in the freeze concentrate, and the protein, by not crystallizing, will dramatically raise the viscosity of the freeze concentrate. And as the viscosity of the freeze concentrate goes up, the diffusion of the trihalo's molecules is going to be slowed down. And if those molecules cannot diffuse, they cannot crystallize. That's why this crystallization inhibition is something that you see very commonly. It is not unique to a protein. Most proteins exhibit this behavior. And higher the protein concentration, stronger the effect, because the viscosity goes up even more in the freeze concentrate, and diffusion is pretty much suppressed. That is one of the mechanisms of crystallization inhibition.
Great, thank you. I think there's one more question here coming, and I'm not sure if I understand it 100%, but the question is how PXRD analyzing under cooling samples. Maybe the person is wanting to understand how the PXRD is done, or PXRD analysis can be done under low temperatures or by cooling the samples.
Yeah, I understand the question, Anurag. At least I think I do. I'll answer it, and then if not, we can go back. So essentially, if you have a low temperature stage, our stage is cooled with liquid nitrogen. You can use helium-2. Mechanical cooling accessories are available. So essentially, you have a controlled cooling stage. You put your solution or sample in the stage, and then you start to cool it. So you can control the temperature as a function of, you can control the cooling rate. You can keep it 1 degree a minute. You can keep it 5 degrees a minute. So because of the cooling accessory, you are able to do controlled cooling, and the sample is maintained at the temperature that you want. It is very much like a hot stage X-ray. In this case, you are just using a cooling accessory to cool the system. So these accessories are now available. One famous brand is Anton Par, but there are others too.
Sure. Great. I don't think there is any other question, but probably there'll be some more questions coming in due course. In the meantime, if I may ask just one question about, there's one more question. So let's finish that first.
Okay.
Oligosaccharides molecule crystallization is possible. So that's probably a statement as a question, whether that is possible.
Yeah, can there be a little bit more specific? I don't know what the question is referring to. How big is it? How big is the oligosaccharide? So general rule of thumb is that as the size starts to go up, the resistance to crystallization goes up. Of course, it's not universal, but in general, as you start to go into the thousands Dalton molecular weight range, crystallization becomes more and more difficult, but it can happen.
Yeah, okay. Yeah, and the person is also indicating you addressed his question well.
Oh, okay. Okay. Anurag, you had a question?
Yeah, I was just trying to understand, Suri, the direct or indirect applications and where this is going to help in development and or any examples, live examples, real examples in the commercial products, or where it is going to make an impact.
Yeah. So the whole issue of buffer salt induced destabilization has been reported for about the last 20 years. So what people have observed is that under certain conditions, because these are all legacy products, people have used buffers, particularly phosphate buffer saline is very popular in the diagnostic industry. So they don't want to change the buffer because then you got to do a whole set of studies. So they use the buffer at a low solute concentration. What they see is denaturation. The product degrades, and it degrades in a random fashion. Doesn't happen all the time, but certain batches happen. Now, the solution to that is really trying to prevent the buffer salt crystallization, and the way to do that would be if you can add a non-crystallizing solute, such as sucrose or trehalose. The other area where people have found this problem, Anurag, is if you are developing platform formulations. So you've got a platform of excipients that you think you're going to use for all your formulations. And in one case, you have a protein concentration, 1 mg per mil. The second case, you have 10 mg per mil. Very different behavior.
Yeah.
Because the protein is an order of magnitude difference in concentration, and nothing intrinsic about the protein, but the fact that it can modulate the crystallization of the buffer plays a big role in destabilization. The other example, about 10 years ago, Genentech is a company that has heavily used trehalose. They pioneered the use of trehalose. And what they observed was that there were aggregation in certain protein formulations after freeze thaw. And they were not even freeze drying it. They were just storing it frozen for a year or two, long-term storage before they formulated. And whenever they saw protein aggregation, they also saw trehalose crystallization. That's how this link became established. So though we are talking about freeze drying, you know this better than I do, when they make these proteins, the campaign is going to be for a long time period, right? The protein is going to be stored frozen for a long time period. And during the frozen storage, if trehalose crystallizes, it can lead to destabilization. So that's particularly because the frozen storage is for a long time, the crystallization will be slow, but it can happen over a longer time period.
Okay, great. Thank you. Well, I think at this point of time, that's the list of questions we had. So may I suggest that we move on, and in the meantime, if there are more questions, we can flash it later during the course.
Yes, and Anurag would like to just mention that I think it's a little late for Raj for this talk. Already it is close to, I think, midnight for him. So would like to felicitate him, and maybe during the panel discussion, if there are further questions, we will post those queries to Raj. So would like to felicitate him, Anurag.
Absolutely, sure. Yeah, so thank you, Suri, for your nice presentation, and appreciate you taking time this late evening hour, or rather midnight hour, to make yourself available and present. Very much appreciate. Thank you on behalf of APA and the organizing committee, and I'm sure participants have gained some new insights through your presentation in this area. Appreciate.
Thank you very much for the chance. Yeah.
Thank you,
Yeah.
Raj. And in fact, would like to just re-emphasize that we really appreciate you taking your time out and attend this and give a talk at late in the evening hours. And we would also, on behalf of committee, would like to in advance invite you next year. Hopefully, we can have you in person when we do our APA India 2022 meeting, physical meeting. So with that, once again, thank you, Raj. Thanks for a very interesting talk. Thank you.
Thank you. Bye-bye.
Bye. Okay. Shall we move on, Deepak?
Yes, please, Anurag, go ahead.
Great. So after this, an excellent talk by Suri, let's move on to the next speaker, who is again distinguished and established in the area of drug excipient compatibility. And I'm talking about Dr. Ravi Shah. He's professor of pharmaceutical analysis at NIPER Ahmedabad. He is alumni of NIPER Mohali, earned his master's and PhD degrees from the institute, NIPER Mohali. And then he started his career with Biocon BMS Research Center in Bangalore, where he actively researched in the area of new drug development and contributed in IND filings in that organization. Then subsequently, he was part of Dr. Reddy's as associate director, and he was heading the analytical R&D in Bangalore facility, where he was responsible for supporting the generic drug development program, where he contributed in number of DMF ANDA filings in US and Europe. He is he has extensively published in peer-reviewed journals, international journals. He and his research team currently works in the area of various analytical topics, including, for example, impurity profiling, drug excipient compatibility, dissolution topic, impact of dissolution and CQAs through structural changes in biopolymers, biomarker qualification. So wide range of topics, and he's very actively involved in the area of analytical sciences and regulatory challenges, solving those. So with that brief introduction, may I give it back to Ravi for starting his presentation. Thank you, Ravi, for taking your time. Over to you.
Thanks, Anurag. Let me try. So my presentation is visible?
Yes, we can see you are audible.
And would request Ravi if you can switch on your video as well. So yes, so we can, great. So you're now ready to go. Thank you.
So very good morning and good evening for all the participants. And on today's topic, we are going to discuss drug excipient compatibility studies and what industry is currently doing, what are what are other aspects of a drug excipient which is ongoing. So for few of you, I would like you to first understand what is drug excipient compatibility studies. A drug excipient compatibility study is nothing but an assessment of a pharmaceutical incompatibilities, which means changes in a physical, chemical, or therapeutic property of a dosage form resulting from interaction of a API and excipient. So generally the API and excipient interact, and most of those interactions is for stabilization. However, certain aspects are non-desirable, and Professor Suri, he connected on certain physical aspect of a crystallizations followed by a destabilization of API. So there can be a physical, chemical, or a therapeutic property. Generally, a therapeutic property is not there. It can happen or associated with a chemical or a physical instability. Say for an example, drug interact with the excipient and make a drug excipient interaction product, which can be considered as the impurity. And that impurity can have a change in therapeutic properties. It can also have a property change like on a dissolutions or a hydrolysis of a drug excipient interaction products and physical interactions and so on. So now let's understand what regulatory says. ICHQ 8 says the compatibility of a drug substance with excipient listed in 3.2.p.1 should be evaluated. And that's all. Regulatory doesn't say how you are going to do a drug excipient compatibility study, but ensure that you are covering all possible incompatibilities and reporting them. Now, this is like slightly changing the game. If you are working on a generic industry and a RLD or innovator product has certain excipients, and because of IP issues or because of better drug profile, you are changing certain excipient. When you are changing certain excipients, which are not used by innovator company, and if you're not done proper drug excipient compatibility studies, you are going to receive a comment from regulator in general, either in the form of a DRL or in the form of a CR letter. And the questions will come how you have done the drug excipient compatibility studies, or even they can refer certain paper and ask to do a drug excipient compatibility studies in a thorough way. And that type of scenarios in few of the cases we have seen when we were in the industry. Now let's understand what is the scientific necessity for drug excipient compatibility. So I'm going to talk more on the chemical instability in my subsequent slides, and mainly for the products of a OSD or the products of related to LIO. So let's consider, say this is the tablet, and this is like an illustrative figure where you can see a yellow color is excipient and the green color is API. Now, during a storage, there is the probability of humidity come into the picture. It can also come into the picture when the processings are there of a drug formulations. And as a result, the excipient and drug get dissolved and make a supersaturated type of micro environment where the excipient drug are dissolved, and because of a temperature or a light during a storage, the drug and excipient can act as a reactant, and energy is sufficient to make a product, and those product is nothing but a degradation product or a drug excipient interaction products. And as a result, it can show degradation. This is an illustrative figure. Please do not consider this will be like an actual figure. Okay. And impact can come from a water, pH, dissolved oxygen or air in this microenvironmental pH. And this is basically showing a drug excipient incompatibility. Now let's understand what is the existing practices in industry. In most of the industry, the people mix the drug and excipients, generally through physical mixtures. In certain cases, add a minimum amount of a water if the OSD product is having a wet granulation, or we are thinking about a suspension product. In case of a lyophilization product, people first dissolve and get it lyo and then put on stability. Or in the injectable products, people can put a vehicle, which can have a mixture of a preservative and the vehicle, and then keep in a stability chamber. Generally, at the accelerated stability conditions, in few of the cases at a higher temperature. And then sampling will be there at 15 days, 1 month, 3 months, and so on. And then put on analytical method. And again, please remember, this is a drug development stage, so analytical methods are not validated. They are like a fit for purpose type of method. And then degradations get generated, and in most of the cases, the degradations is lesser or less than 0.5 area percent. And if it is less than 0.5 area percent, most of the company or most of the scientists report the drug is stable. And at a later stage, when on an actual accelerated 6-month stability or in a long-term shelf life stability, a peak started coming, and then there will be like a rush. What is this product? Will it be possible to file a ANDA, or will it be possible to decrease the shelf life period and still file and so on? These all things comes at a late stage if a preliminary study has not been done properly. And in a preliminary study also, there are a lot of challenges and a root cause. Let's start talking about what are the challenges. The first challenge is the segregation of API and excipient. When you try to mix the API and excipient for a drug excipient compatibility studies or putting on accelerated conditions, the particle size, shape of the API and excipients are different. Whatever way you do a physical mixture, you need to be very, very cautious about mixing the API and excipient, because the segregations can happen. I've shown in the figure that the segregation can lead to a less exposure, or the surface is not exposed for an API with excipient. And in a previous slide, we have seen if the microenvironment is not generated, the probability of a degradation decreases. So the close contact between the excipient and the drug is necessary. And we need to be very careful for that. So people try to overcome with that by making the slug. So first they take a drug and excipient and make a slug. And then putting maybe a water and make it a suspensions and before putting into the accelerated conditions. The second problem is unknown risk associated with a lot of API. So when I say a batch or a lot of API, is in many of the cases, if you are working in a drug discovery phase or a IND or a phase 1 or phase 2 clinical stage, the chemistry group is giving the API, and at that time, and they have not concentrated on the particle size of the API. One batch comes with the larger particle, another batch comes with the smaller particle. Sometimes the API comes with a needle shape, another time it comes with the rhombic or a cubical shape, and so on. And as a result, when the API particle size is differing, or even in certain cases the polymorph get changed. Say for an example, as a formulator, you are taking API from outside. You may be taking from the company of API supplier just for a namesake. If you are taking from a lupin, it's giving a polymorph M. If you are going with MSN, it's giving a polymorph B. and so on. But the drug excipient compatibility study, you have started much earlier. And later on, the form of the API get changed or vendor get changed, and you do not have a time to do a drug excipient compatibility studies again. So needs to be very careful of understanding the API property before we do a drug excipient compatibility studies. So as a potential solutions, it's always advisable to micronize without impacting significantly on morphology. So at least with the conventional way, like a pestle and mortar, you need to grind the API as much as you can, make a bigger lot, and from that lot you should be using for drug excipient. The another problem is again the duration and the waiting period of our results. I have seen in certain companies drug excipient compatibility studies goes for 3 months, and the drug development cycle is extended. If you are working on a generic industries, you have maximum of 1 year. In 1 year, you need to complete your entire development and ready for filing. That is like the expectation. Now, if you want to do a drug excipient compatibility studies in quick enough time, it is extremely important that you have a right sample preparation as well as the right stress conditions. So all aspects, it's extremely important to increase the surface area. And a stress condition also needs to be optimized. In many of the cases, it's not possible to give a stress conditions at a 40 degrees C and 75% RH for OSD, or for injectable 25 degrees C and 65% RH. It's not feasible because you need to wait for a longer duration. And it's always advisable to increase the temperature and the relative humidity. However, there are disadvantages because you may get unrealistic degradation product. However, you need to balance between the duration and the drug excipient compatibility studies. Another problem is from analytical aspect also. Your method is like a fit for purpose. And you can see in the blue line, there are certain impurities which are very smaller, which is less than even 0.1%, or less than even 0.05%, which is a reporting threshold. And you need to ignore, because that can come from any of the analytical error associated with that. But if the degradation is sufficient and the difference between the samples are greater than 0.2 or 0.5%, it is very easy for you to select the appropriate excipient. Please understand that we do a drug excipient compatibility studies to select the best excipient possible, which has a minimum degradation. So you are going to screen excipients and select the appropriate one. Maybe say a diluent. You have a multiple diluent like a mannitol, lactose monohydrate, and so on. And you are going to select one out of that. And when you are going to take a decision based on a drug excipient compatibility studies, and the HPLC profile or the percent degradation is going to give you output, it is always better to have a discrimination between the excipient, and those discrimination has to come if the degradation product is in sufficient quantity. So please be mindful whenever the drug excipient compatibility study goes. Many a time people reject the excipient because of the analytical error, and even though insignificant difference is there in compatibility studies, it is shown as a significant difference. So you should be mindful of that. So let me tell what will be the ideal condition. So the ideal condition can be for early drug excipient compatibility studies. You have excipient, so you can see in a picture excipient having a 20 or a 50 micron particle size. And on those excipient, if you can put the API in a much smaller particle size, less than 2 micron, then the effective surface area which is coming in contact will increase significantly. And the second thing is a stress condition can be put as an accelerated stability conditions rather than putting at a higher temperature, like 80 degree or a 60 degree or a higher temperature than the actual requirement of the dosage formulation. So this is like an ideal condition, means our group is working here and make a, a prototype which is feasible with the different industry to follow by certain modifications of the instrument and other things. So in a subsequent years we will try to give solutions for these challenges. Now let's move quickly on reported approaches for drug excipient compatibility studies. I have taken the examples from the literature where either the industry is widely using or published by the industry, or they are like a general ways. So the drug excipient compatibility studies or interaction studies started long back in 1983, and this was like a early paper where the drug and excipient were mixed, added 10 to 20% of a water, which is weight by volume, and then put at a 55 degrees C for different hours. So in this paper, it was studied for 511 hours, which is approximately 3 weeks. And then the samples were analyzed through HPLC and DSC. But here also you can see the water was added and put at 55 degrees C. The only disadvantage in these cases can come because the hydrolytic degradation products or hydrolytic interaction products will be prominent because we are adding a water directly. Now, coming back on a similar concept, Sarajuddin and al. from Bristol Myers Squibb published a paper in 1999 where 20% of a water was added in closed conditions put at 50 degrees C. However, they have also put a drug and excipient in open conditions and exposed to 50 degrees C and 75% RH. And try to analyze the samples in 1 to 3 weeks. Coming to the another approach, mainly from the GSK, where they have put an assembly where they are using a stem tube. In the tube itself, there is a separate chamber. In one chamber, a drug and excipients are mixed, and outside there will be a water. It will be heated at 80 degrees C for 1 hour, and there will be a condensing of a water also happens. And then they are analyzing using a HPLC. However, there are reports which says that the 80 degrees C, the drug or a dosage formulation is never going to get exposed. So this will generate a lot of unrealistic conditions and not desirable. Again, from Hoffman Roche, they have published a paper in 2005 where a high throughput screening type of platform was developed, where in a 96 well plate, the API and API excipients were mixed and kept at 40 degrees C and a 50 degrees C with the 10% and 75% RH. Mainly this type of approach is used for drug discovery phase. However, when you decrease the sample size, the appropriate mixing will be difficult. And as the appropriate mixing will be difficult, effective interaction or effective surface area contact between the API and excipients are not possible. And the reproducibility of the data will be at stake. So people should be mindful on selecting the amount of API and how much amount of excipient. So the physical mixing is possible if you are looking for a physical mixing type of approach. There are certain approaches which is widely used for early assessment. So what researchers do is they take a drug and excipient in a DSC pen and run a DSC and try to see whether is there any change on an endothermic event for a lone drug and when the drug and excipient were mixed. So in a layman language, is there any possibility of a melting point get changed when the drug and excipient have been mixed, which can indirectly says that there are a probability of some interaction. And it is very difficult to tell that that interaction is bad or non-desirable. Most of the cases, those interactions are useful also when you are working on certain cases like amorphous dispersion and so on. So just considering the drug and excipient interaction, people try to understand the incompatibility, and there are a lot of possibility of false positive and false negative. Similarly, there are a few papers where instead of a DSC, people try to understand with the IR and PXRD. But again, researchers should be mindful about the possibility of that interaction. It is desirable or not desirable. Now, coming back, what industry or researchers should do. So we have proposed the approach where we are making a drug and excipient, mixing within the GC vial, and insert another chamber where you can put a water, which can stick to the GC vial. Close the cap, put at a 60 degrees C. So when you put at a 60 degrees C, basically the water is going to get evaporated and going to try or make a relative humidity around 95%, which is the saturated relative humidity at 60 degrees C. Moreover, when you do that, there won't be any condensations because the entire vial, this is done in a GC vial, so the entire GC vial was kept at a 60 degrees C oven. And it is not possible with the stability chambers to get a 95% RH. It is practically very difficult, and none of the supplier currently in India or abroad can give a 95% RH with 60 degrees C as a stability specifications, which can run for longer duration. So in this case, the easiest way is the water which is going to get evaporated is equivalent or sufficient to make a relative humidity near to the saturation, and then analyze the samples in HPLC with 1 week or 2 week. In our lab, means our team has done for 36 formulations such studies and compare with the conventional way, and I'm going to show only one case studies with one formulation and only in one condition. So this is the case study here where the valsartan, one of the Indian formulation was taken, and you can see in that the formulation was kept on a primary packaging as an intact tablet in open conditions in a crushed way at a 4075, and added a water 100 microliter in 50 mg and put on a open conditions, which is like generally what people use and put at a 40 degrees C and 75% RH. However, in the proposed way, when we have put at a 60 degrees C with approximately a saturated relative humidity, which is 95%, and they were compared. Please also note that the drug shown a degradations in 1 month and 3 months for all other conditions, which is less than 2%. Whereas in the proposed approach, a degradation was 7% within a week and 11% in week 2. So this was very clear, which says that degradation products are forming in much early stage than the desirable. Now the question comes whether those degradations products are covering all the products or converting to a secondary products. So this is like a graph where a different impurities were mentioned. So each color is giving a impurity, and there were total 8 impurities found as a total. And you can see there was no such instances where that impurity or a degradation product was not observed in other conditions. So whatever has been observed in a stress conditions, all possible degradations are covered. And obviously the amount was also higher. So from the analytical aspect, it was much easy for us to compare. So I will just try to summarize that there are lot of possibility of a drug excipient interaction product type of studies, and there are a lot of challenges associated because of false positive and false negative results, and there are requirement of trying to have certain platform type of technology. So our team is working, and the doctorate or a PhD scholar Sonali is working on this area. So with that, I would like to thank for your patience and hearing. So I can take up the questions if anyone has.
Yeah. Thank you, Ravi. Wonderful talk. I'm actually looking at the chat box as well as the Q&A. So there are no questions. So I definitely have one or two questions in the meantime, if people would ask. So if I may go ahead with my question. So one of my question is, Ravi, there are a number of ways these studies can be designed. And since there is no regulatory framework defined, the flexibility and freedom is given to the scientists and the teams to design the studies for their fit to purpose. And the question is, while designing these studies, you can actually do in so many number of different ways. The results can also be dependent on the way the study might have been done. And always the risk of false positives or false negatives. So do you have any kind of a decision tree or a structured way of identifying how can you segregate and discriminate any false positives and false negatives, which otherwise, if taken as is, then could lead you into wrong directions of either selecting or rejecting the excipients that you would want to utilize in the formulation. So any thoughts and any work that has been established to be able to achieve that objective?
Yeah, so Anurag, very right question. And actually the industry is facing a similar type of problem where they are rejecting the excipient based on false positive results and so on. So I have put on the slide and try to interact or try to understand how it can be done. So let's understand first question. In what are the cases we can get a false positive and false negative result? The conditions of a stress condition means basically a temperature. If it is too high, say for an example, in one of the paper, GSK is using a 80 degrees C, and my storage conditions maybe a 25 C, and my formulation is not going to exposed anytime at 80 degrees C. And there is a probability where the excipient which I'm using, it has a TG maybe around say 70 degrees C, or at 80 degrees C, my excipient, if it is a monohydrate, it can convert to anhydrous. So basically the property of an excipient and API can change in those stress conditions, maybe at a higher temperature. And that is major or a major reason for having a false positive and a false negative results. In our study also, we have seen at a 60 degrees C, a degradation product is also increasing, and obviously the amount is also increasing. And there are probability where in real scenario, those degradation product may not happen. So in an ideal condition, so that's why I kept this slide, and I hope everybody can see. In ideal conditions, the stress condition actually should be accelerated stability condition. Say if it is a 40 degree, 75% RH, you should be doing a drug excipient compatibility studies actually at a 40 degree and 75% RH. But if you do that, you need to increase the surface area contact between the excipient and drug. So our team is currently working and making a instrument, and we have like already on the patent stage. What we have done is we have made the API particles lesser than 2 micron particle, or most of the cases 1 micron particle size, and put those API directly on excipient. So the effective surface area for the contact between the API and excipient have been increased multifolds, which can cover or which can compensate on a duration and a stress condition. So the objective here is ideally the drug excipient compatibility study should be done at accelerated stability conditions. And accelerated stability condition is well defined in ICH and everywhere, where actually the product is going to get exposed. But it needs to compensate with certain other things. Now, if you see the scanning electron microscope very carefully, you can see in a 1 micron particle size also, they are concentrated areas. So when we have sprayed, it was an amorphous material, and those amorphous material was started converting to a crystallized material also. And you can see a small patches, which is the actually crystallizations within the amorphous surface. So we are working on that. But Anurag, you are right. It is extremely essential for all of us to understand whether we are using a right stress condition so that the false positive and false negative do not come. So we have done, means we have not yet published the studies, but with the 60 degree C and so on, we have like done with 36 formulations and try to compare with the possible understanding on drug excipient interactions, considering the false positive, false negative, and so on.
Sure. Thank you, Ravi. I think there are a number of questions which are pouring in, and we probably would need more time to address several of those, if not all. Why don't we, I would propose that we go to the next talk, and during the panel discussion, we can cover all of those. Maybe there'll be more similar questions which could be bunched together and addressed in one shot.
Yeah, make perfect sense.
Well, thank you. Yeah, thank you, Ravi, for a wonderful presentation. So let's move on. And sorry, I think Deepak wants to provide the certificate of appreciation. So thank you, Ravi, for the talk, and on behalf of the APA organizing committee and all the participants, I thank you for taking time and presenting a wonderful topic to the audience.
Thank you, Ravi. And as Anurag mentioned, we'll take up the other questions during panel discussion. So Ravi is going to be there in panel discussion as well. So request all the participants to keep posting your questions, and at the end of the webinar, in the panel discussion, Ravi would take up those questions. Thank you, Anurag. So let's proceed further with the next talk.
Yeah. Great. So let's continue this journey of learning about this excipients and extractable leachable theme topic today. And the next talk is on the extractable leachable studies, and that is being delivered by Mohinish Sahai. He is currently working with Baxter India. He is a pharma R&D professional with about 15 years of experience in analytical sciences. And his main area of research and emphasis has been the extractable leachables. As I said, he is currently employed with Baxter Pharmaceuticals India. He is working there as tech lead and focusing on this specific extractable leachables area in the organization, where he is defining the E&L strategies, outlining the plans, managing execution of the studies as the study director. He provides support across various programs wherever the need is for the regulatory sections, and helps in answering the queries and approval process for the drugs and drug products. His other experiences in the pharmaceutical dosage forms and container closure systems for small and large volume parenterals, transdermal patches, etc. By education, he is a postgraduate in chemistry and business administration, and he's currently pursuing doctoral research degree in the field of genotoxic impurities. So with that short introduction, may I give it to Mohinish to take over from here and present. Mohinish, over to you.
Yeah, thanks, Anurag, for giving this opportunity. I hope my screen is visible.
Yes, Moinish, you just have to do the slide share and you're good to go.
Okay, thanks all, and a very bright morning and a very cool evening to all the audience. I, Manish, will be today presenting on a topic which is a recent addition to an analytical field. I'll say it is not too old. It's a young buddy over there in analytical field. It's extractable leachables. So today we'll be discussing about the technical challenges and consideration for extractable leachable studies for the complex large volume parenterals. So for those who are not very much familiar with what is extractable and what is leachable, and you might be thinking that from where does the stuff escape. So let me give you a brief introduction for extractable and leachables. So just like the impurities which we get from a product, API or a drug formulation, extractable and leachables are the impurities which we get from a container closure system. Means your the glass vial, the rubber stoppers, whichever we are using to store our drug formulations. So the impurities which can come out of those are known as extractable and leachables. So what is extractable? So extractables are the substance that can be extracted from a material, device, or system using simulating solvents under the extraction conditions that minimally mimic or exaggerate the actual contact between the material. So in a simplified version, anything which can come out of a container closure system with the help of solvents under the aggressive or a mimicking condition is known as extractable. Now coming to leachable. So what is leachable? So leachable are actually a subsets of extractables. So the impurities or the substance which can come out of a container closure system when the actual product is in contact with it. That is during the storage or during the formulation, whenever the drug product is in contact with manufacturing lines or container closures. So what can come out of those lines or container closures into the drug product are known as leachables. So there are various regulatory agencies, advisory bodies, and industry groups which have given some of the guidelines for this extractable leachables per se, but there is no harmonized guidelines for extractable leachables as of now. So different bodies have their different perspectives. So, but mostly the alignment is with USP and PQRI. Coming to some of the common terms which are used in extractable leachable studies, they are AET. So what is AET? It is an analytical evaluation. So it is just like a identification threshold. If any of these impurities or these substances are going above this level, we have to identify. What is the safety concern threshold? That is SCT. So it is a threshold below which there is a negligible safety concern. So if our substances are below this level, then the safety concerns are negligible. MDD, that is maximum daily dose. So as we all know that it is a maximum quantity of dose which can be administered to a patient in the time interval of 24 hours. So coming to the technical challenges and different product types. So as we all know, one size does not fit all. So there are different container closure system, there are different devices for which these studies are required. Examples of some of them are like meter dose inhalers. So what are the technical challenges which we face when we are dealing with meter dose inhaler during this extractable and leachable study? So for the meter dose inhalers or OINDPs, that is orally inhaled nasal drug products, the sensitivity limit which has been kept is very low. It is near to about 0.15 microgram per day is the safety concern threshold. So for that, exhaustive extraction studies are required so that we can come to know that what is the worst case scenario, what are the worst case substance we can extract out from that device. And because we will be getting a whole lot of components during our extraction, so that will lead to a complex leachable profiles too. So the other challenges which we will be facing over there, that there will be unknowns which cannot be identified because they are at too low level. So in that case, the implementation of a control strategy will be another challenge. Second, coming to topicals, ointments, creams. As we all know that creams are generally emulsions. So when we are dealing with emulsions, we deal with a lot of matrix interference. And when there is a matrix interference, it is very difficult to identify or quantify a particular substance, which is somewhere in ppb levels. So the formulation is the biggest technical challenge when we are dealing with topicals or creams or ointments. So because of that, as it is an emulsion, so the another challenge which will come into picture is that what kind of solvents you are going to take to mimic that formulation. That is to design your extractable study. And similarly, following the part that how your leachable strategy will be, how you are going to remove those interference, how you are going to quantify or screen those unknowns. So that is the third challenge. So this topic is mainly about large volume parenterals. So we'll be talking mostly on large volume parenterals like bags, which will be having the volume of something about 2000 ml to 5000 ml. So for large volume parenterals, the basic challenge is that poorly defined MDDs. So maximum daily dose cannot be defined for large volume parenterals to a much certainty until there is a clinical expertise or based on the clinical benefits, those doses can vary. So because of this high daily dosing volume, the method sensitivity will go down to a too low level. Means we have to keep our method sensitive even to about PPT levels. I'll be showing that in the subsequent slides. So due to this low level of AET, that we have to identify any particular substance or impurity which are coming above, suppose 1 ppb or 2 ppb, you can understand that how many unknowns can be there. So identifying those unknowns is another big challenge. So what is the take home? So take home is that each dosage form has its unique set of challenges, and it is not appropriate to apply the same generic approach to a variety of product types. So here I'll show some of the schematic of critical E&L activities, how it proceed, what are the steps. So these are the broad steps which are there in E&L studies, like CCS material selection, extraction studies, and then followed by leachable studies. So during our container closure material selection, what we see generally. We see what is the cleanliness of the material, what is the stopper coating. Why stopper coating is important? Because if your stopper is coated, then the chances of migration of your substances or leachable comes to a very low level. Then we have to see that what is the maximum daily dose, and based on that, our analytical evaluation threshold will be defined. Post that, we will be seeking for extraction conditions, that what is the extraction conditions that we need to see. So as Dr. Ravi has also spoken before, that during the drug excipient stability studies, we have to mimic the condition instead of going too exaggerated. So the same concept applies over here also. We can go with a much accelerated condition or aggravated conditions, but is it really required? Because our drug product is not going to expose to that such worse conditions. So those are some of the things which we need to take care during planning our extraction studies. After extraction conditions, the method sensitivity plays a vital role. We have to keep our method sensitive at least up to the level of AET. Generally, we go with a 50% of AET, but your method should be at least at the AET, it should detect the compounds. Post that, we'll go for a toxicological assessment of all the substances or all the extractables which we have found, and we have to identify the unknown extractables possible. Once our extraction study is completed, we go for a leachable study. We have to design our leachable study. We have to see that whether we want to go for a target leachable study or we want to go for a screening study, and that is based on the drug product formulation and the interferences and various other factors which I'll be talking in the subsequent slides. What is the take home? So the take home is that analytical evaluation threshold is a critical input when designing a strategy that addresses the toxicology associated with patient exposure to leachables originating from a container closure system or manufacturing process. So what are the analytical challenges? As we know that PQRA PDP working group has proposed a safety concern threshold of 1.5 microgram per day. This is in the case of ONDP is 0.15 microgram per day. And that is regardless of treatment duration, whether it's weekly or monthly or yearly or lifelong. And we know that SCT is used to calculate the AET. So generally SCT and maximum daily dose is used for the calculation of AET. So SCT divided by maximum daily dose will get our AET. And for analytically meaningful units, it should be in microgram per ml or microgram per stopper. And you can see that as MDD is inversely proportional to AET, so the lower the AET, the greater the analytical challenge. That means that higher the maximum daily dose, the lower the AET, and the greater the analytical challenge. And sometimes really it is not possible to align with the AET, even using the latest state of instrumentation. So what's the take home? The higher the MDD, the lower the AET, and greater the analytical challenge. So what are the challenges associated with defining the MDD for some of the large volume drug products? So if we take a case of sodium chloride injection, like USP in a Viaflex plastic container, we can see that the dose of sodium chloride is not fixed. It is highly variable and dependent upon the patient needs. For example, in hypovolemic shock, an initial rapid infusion of several liters of fluid is required, and then additional boluses is required. So there is no fixed MDD. We cannot say that the patient will require only 2 liters of sodium chloride or 1 liter of sodium chloride. It depends upon the clinical administration. It depends upon the situation. So if your MDD is not fixed, then how you are going to calculate your AET, and then how you are going to proceed with your extractable and leachable study. So that is a big challenge. What's the take home? The MDD of some of the products cannot be defined, resulting in the need of clinical expertise and assumptions to be made. So in that case, the best case is that we have to consult our clinical experts and see that what is the more clinically relevant MDD we can take. And based on that, we can justify and proceed with our studies. Method sensitivity. The the other big challenge? What are the realistic expectations out of it? So if you'll see, as I already told, that as the MDD will increase, your AET is going to go down. You can see if I am taking a safety concern threshold of 1.5 microgram per day, and my maximum daily dose in ml is 10 ml, I'm getting something good amount of 150 of my sensitivity. I can easily develop a method on that using the latest state-of-art instrumentations like HRMS or Orbitrap. But once you go to the higher MDD volume, like the case what I discussed about sodium chloride, you can see that the ppb level is about 0.3. My gosh, that is 0.3 ppb, that is 300 ppb. So do you think that this type of sensitivity can be acquired? Yes, if it is a clean aqueous solution, definitely we can achieve that. But what if there is a presence of propylene glycols or the toluenes that creates a lot of matrix interference? In that case, it is really difficult to achieve this sensitivity. So what is the take home? So yes, the AET can be achievable with the state-of-art instrument, but it depends upon the sample and sample preparation. Or else we can go for a highly optimized targeted. What are the factors that need to be considered for matrix sensitivity? So performance of an instrumentation, it depends from vendor to vendor. Definitely, if I have developed one method on suppose A instrument, there will be some difference in parameters in B instrument, and I need to modify my method according to that instrument. So those are minor differences. We won't get a huge differences from say instrument to instrument. The main thing which affects the method sensitivity is sample preparation. I already told, if you have a lot of matrix interference in your sample, then definitely you are not going to get your analysis correct, because you cannot get most of the peaks which are at very low levels, and it can be masked by those matrix interference. So common techniques which we use are like static headspace for volatiles, liquid extraction for semi-volatiles. Going to advanced techniques now, we are using dynamic headspace, SPNE, and there are various other techniques which are key, which can help us to go to a much lower level. The choice of internal standard. So it depends upon the organization that what is their strategy, what is the choice of their internal standard. We can take an internal standard with a very high response factor, and from that we can go to a low level of sensitivity, even to a PPT level. But does that resolve the issue? Does that make sense? Because most of our leachables which are going to come or extracts are going to come might not have those high response factors. So it is very critical how to select the internal standard to avoid overestimation or underestimation of sensitivity. So how generally we define sensitivity? We can define on the basis of concentration of internal standard. We can define on the basis of lowest estimated compound that is detected with a signal to noise ratio greater than 10. And we can define with the mean theoretical limit of detection determined from a diverse range of analytes. So we can put diverse range of analytes, a mix of about 10 to 20 analytes, and that mean theoretical LOD we can take as a sensitivity of matter. What's the take home? The take home is that the sensitivity of matter varies from one compound to another. And the generic approach cannot be applied. Coming to the extraction condition, as I already told, that we need to be very much peculiar. We need to be very much focused at what we actually want. Do we want the worst case scenario where my product is never going to be exposed? Or do we want the simulating condition? What is the more realistic environment for my product? Definitely there is an advantage of replicating the interaction, the time, temperature, solvents. We can increase the temperature and time to a higher extent to get the worst case scenario. But definitely that depends upon the strategy, what you want. You want the worst, or you want the actual reality, which is going to come, which is going to extract from the container, which is going to reach from the container to our product. So Solvent choice, like we have to see whether if my product is aqueous. Definitely, if I'm going to take hexane as my extraction solvent, it doesn't make any mimicking characteristic to my drug product because it is aqueous. So whatever it is going to extract will be in aqueous form. So I'll be most probably taking acidic and basic pH buffer for the extraction. So solvent choice also matters a lot. What's the take home? So take home is that be clear on the intent of extractable study and select conditions with that intent in mind. Predict leachable profile or chemically characterize a material or CCS. So what's the impact of AET on the toxicological risk assessment? So as I told that even with the latest state-of-art analytical instrument coupled with mass spectral libraries and structural elucidation techniques, we cannot identify all compounds at such a low levels of PPT or PPP. Some of the extractables are not commercially available, right? Rubber oligomers, which are the integral part of a rubber stopper. It is made up of these oligomers, but those standards are not available. What to do in that case? We cannot go for PrEP HPLC or NMR because the amount of these leachables and extractables are so low that cannot be collected. So for those extractable leachables that cannot be identified, it is not possible to assess patient risk using the analytical data. In that case, what shall we do? When we cannot identify, what shall we do? Then the role of biocompatibility data come into picture. So the biocompatibility data that is in vivo, in vitro on that extracts justified as being representative of leachable profile. And that could facilitate establishing patient risk versus clinical. What's the take home? It's unrealistic to expect an analytical chemist to be able to identify every extractable or leachable compound at ultra trace levels. So we have to consider the biocompatibility data to justify that. So what are the factors that need to be considered for leachable study? As I already told, the drug product matrix index, as it can inhibit my sensitivity. We have to decide whether we go for a screening method or a targeted method. If for a targeted method, if there is known extractables which are coming out of a container closure system, I can go for a targeted method and analyze those known extractables during my leachable studies, what among those extractables are coming out in my drug products. Based on the toxicological assessment, based on the extractable profile, if it indicates risk, we can consider the benefit of including multiple batches and time points to minimize the risk. So what are the opportunities for alignment and industry standardization, where the pharmaceutical industry as a group can align and follow a particular thing that can justify the regulatory agency? The one field where the alignment should be there is definition of sensitivity. To recognize the sensitivity of a screening method applied to extractable or leachable study, it is only an estimate when comparing against the AET. So sensitivity is only an apparent number when comparing against the AET. So we should consider standardizing the process to estimate the sensitivity by using a diverse range of compounds. Coming to maximum daily dose, recognize that some large volume parental drug products, they don't have MDD or they are variable. So can we go with the risk by capping the MDD at 2 liter as defined in ICH-Q3? So in ICH-Q3, it is clearly mentioned that if your MDD is above 2 liter, you can cap it to 2 liter. We have to see whether the regulatory agency accepts it or not. What to do about unidentifiable extractable and leachables? So as I already told that we don't have the magic instrument which can identify all the extractable and leachables coming in the compound due to low level of AET which is there in PPT and PPP levels. So in that case, we have to see what are the clinical benefit by using the in vitro in vivo data and by using the current state of art instrumentation. So that's all from my side. Thank you very much for listening. I'll be happy to take any questions.
Yeah, thank you. Mohinish, would request you if you can stop sharing the screen so we can showcase a video. And by and large, you were completely audible and it was very clear. Only when you slightly move away from the mouthpiece, the voice was going down, but it was pretty audible. So thank you for a very interesting talk. Over to you, Anurag.
Well, yeah, so I echo word of thanks for presenting. And I think there are questions coming in. Should we just take all the questions together later on during the panel discussion, if that is okay, Monish? Are you going to stay back till the panel discussion? Okay,
Yeah.
wonderful. So just make it more efficient, and we can take all the questions later on. Deepak, you want to present the certificate? Can we move on?
Yes.
Yeah. So just formally, once again, thanking you, Monique, for taking time on a Saturday morning and presenting. I'm sure the audience is looking forward to the panel discussion and they have a few questions. I appreciate you staying back.
Thanks, Deepak.
Excellent. So let's move on to the next and last talk of today. And, this is such an area, the excipients, compatibility, extractable, leachables, etc. And the discussion would be incomplete if we don't cover the regulatory aspect and regulatory perspective. So that's where the next presentation fits in, the theme today. And we have an excellent speaker, Dr. Amjad Anwari, who is associated with USP India as Director, Small Molecules and Excipient Science Division, and based out of Hyderabad. He has a PhD degree in chemistry from University of Mumbai. He has around 19 years of experience in pharmaceutical industry, R&D, QC, where he has contributed across several products programs for NDA and ANDA filings. He's a member of IPEC, the Indian Pharmacopoeial Committee, and he also holds FDA approved certificate for chemical testing as well as instrumental testing. With that, may I hand over to Amjad for starting your presentation. It's nice to see you again, by the way.
Same here, Anurag. Thanks for that warm welcome. Thanks to Deepak for having me here, and thanks to APA for making this possible, sharing this platform with all of you. So good morning, all the participants, and I'll share my screen now, which is here. All right. So I hope it's in presentation mode. I'm visible and audible as well, and good to go.
Yes, Amjad, you are good to go. Thank you.
Thanks, Deepak, for that confirmation. So yeah, welcome everyone. Good morning, good afternoon, good evening, wherever you are located in the global and in the virtual world this time that we are going through. So in this next 20-25 minutes, what I will try to cover is taking you through the organic impurities in the pharmaceutical industry, USP perspective. And the idea for this talk is to, of course, the main center of attraction you will see will be on reporting thresholds, which plays a very vital role in the world of impurities. And since this whole theme is all about challenges and the opportunities, because we know, right, that there are challenges in our day-to-day life and in pharmaceuticals, but along with challenges, what comes is the opportunities for us to make it better. So we will discuss on all those fronts, but before going towards the reporting threshold directly, I would like to make some background with respect to some references coming from USP, coming from FDA, coming from ICH. So that will set a really good tone, and then we can have a really good overview and a fair discussion when we'll start covering reporting threshold. And of course, in the last, some few glimpses on excipients front as well with respect to impurities. So let's get started. As a general overview, as you can see here, it's like, as we see in the pharmaceutical, that how the process goes. And what we try to cover here is like the completely from the raw materials or the starting materials when we start our synthesis, whether it's route 1, route 2, or route 3, right from there till the drug product, how this journey goes and where all we see impurities as there in all this process throughout. So when we talk about those synthesis, which is the very first step where we start with all those starting materials, whether it's A or B, and taking those solvents and catalysts at particular time or temperature at given pH to ensure that we are forming that intermediate C. But doing that, of course, it's not only C that we get, but there are also some impurities, some byproducts that comes along with it. And it follows when you even take those intermediates to the stage of drug substance. It's not only the drug substance, but we see impurities associated with it coming in, along with the degradation parts, byproducts. So it's all part of the final drug substance that we receive. And the same trend follows when we take this drug substance to the drug product using excipients, for which we had really good discussion since last 1.5 hours related to excipients, so many good stuff about it. So this is where when we reach to drug product using our drug substance and excipients, again, we can expect those impurity coming in as a part and parcel of the drug product, where we have to be very cautious. And of course, the leachables from container or closure, which was really a good talk before this by Monish, where, most of the time we focus on the drug product quality, but also what becomes very important, and it's good to see it's evolving in this today's world, is on extractables and leachables, where we know, right, that packaging is also a partner of our drug product throughout its shelf life. So it's very important to have those perception also in place, that how extractables and leachables are also getting into your drug product, if at all they are, during the shelf life. So what impurities are we interested in? Well, it could be many, right? It could be residual solvents. It could be inorganic impurities. It could be organic impurities. Residual solvents generally covered by USP 467 general chapters. And of course, you can also see those contacts in general notices like 5.60, 0.20, which applies to all monographs. And of course, you can also refer to ICHQ3C for that. There are also inorganic impurities, which could be like, through specific tests for chloride and sulfates. It could be for non-specific tests like ROI, and then elemental impurities, of course, which speaks about limits. And if many of us remember, there used to be a general chapter called 231, which was replaced by later on then 232 and 233, with evolving methodologies and to make it more sophisticated. Then there is organic impurity, which involves so many factors, including chromatography, which is very well defined in USP 621, ordinary impurities, and of course, the chapter on impurities in drug substance and drug products, which is given by 1086. Well, what's the reason behind revising this general chapters of 1086? Because we know, right, as we discuss about those challenges and opportunities, it's always good to keep yourself updated on the trends that is being coming in, the technologies that is being coming in. So this chapter revisions, whenever we do, we take care of all those aspects. It brings in lots of, we could see those challenges like those outdated procedures, lack of clarity. So the possible basis for this revision was also upgradations and ICH guidelines, guidance for industries, ANDAs. So it's a lot of factors comes in whenever this chapter goes under revision. There are really good guidelines or the guidances from FDA, which we see through the MAPS, as we call it more frequently, which stands for Manual of Policies and Procedures, which helps us and understanding those impurity acceptance criteria as part of specifications, then whether it's NDAs, ANDAs, and even BLAs for that matter. And CEDAR, like what we call it normally as for Center for Drug Evaluation and Research, published it in Jan 18, 2018. And then, of course, there was a revision in September 19, 2018. And then, of course, recently we have seen those revision on Jan 5, 2020. So it constantly goes under revision to ensure that it's with the trends that has been observed in the industry. There is also guidance for industry ANDAs impurities in drug substance, which was published in June 2009, and for ANDAs, of course, November 2010 by OGD. So when we discuss about all these chapters by all this 1086, there are a few things which we really have to keep in mind from manufacturers' point of view, the responsibilities that comes in, which are very important to ensure the drug product quality. And that's where all these few bullet points that we try to capture upon is like the manufacturers are responsible for controlling organic impurities in accordance with current regulatory standards. Also, those analytical procedures that were used for detection and quantitation of impurities, it should be ensured that they are verified as well as validated based on the conditions that you are in, whether it's an in-house method, you're using some compendial methods, but it should be ensured that it's verified or validated accordingly. Then, if an individual monograph does not include a procedure for quantifying an impurity, or if there is no acceptance criteria for an observed impurity, it becomes the sole responsibility of the manufacturer to develop and validate analytical procedure and establish an appropriate acceptance criteria. Then the last one that we would like to focus on is on the acceptance criteria for impurities shall be based on applicable guidances or other acceptable scientific means. I would reiterate acceptable scientific means, right? Which is very important to have those scientific justifications for whatever acceptance criteria you're coming up with, you're going ahead with your filings, because those are the areas which ensures that safety has been taken care of as the primary considerations. And it's not just solely based on the capability of the process. Few outlines that has been covered in the chapter is on introduction front, drug substance, drug product, decision tree, which helps a lot at times to see where you are on your process and how it will guide you to move forward. There are a few glossaries and of course, additional sources of information. Then let's have a look at organic impurities and drug substances and drug products, how basically it works, because we were about to start discussion on reporting thresholds. So basically it should be a threshold-based approach, as also described in ICH Q3A and Q3B guidelines. And you should be used for the control of organic impurities in drug substances or drug products, which generates during the manufacturing process or even during the time of storage. There is also a cross-reference to 476 and the glossary section. And 476 is a recently published general chapter which talks about control of organic impurities in drug substance and drug products. In case of complex impurity profiles, right? I'm sure most of us comes across this complex impurity profiles. It's very less that we see a very simple and smooth one, but most of the time we come across those complex impurity profiles. So manufacturers should consider all those alternate approaches, such as the use of those multiple analytical procedures to test for impurities, just to ensure that there is no stones left unturned, to ensure that the test or the data that is being generated are optimum. Similar principles may be applied to set thresholds and acceptance criteria for degradation products in FDA OTC monograph drug products. Most of the time, being OTC, being less of regulations, there is a leeway, we go around, but it's really good to ensure that we set thresholds and acceptance criteria for even OTC as well, which are not discussed in ICH guidelines or FDA guidances. And degradation product in this drug should be reported, identified, and/or, I would say, to be qualified. So there are a few things that has been covered here. It's like on glossary, which is about the disregard limit, which is same as reporting threshold, and it may appear in monograph as and disregard, and mostly you will see it in our older monographs. And in reporting thresholds, when we talk about that, it's a chromatographic test. These are the limits above which an impurity should be reported and should be taken into account for calculating total impurities or total degradation products. Because we know, right, when we work on any analysis, we see n number of peaks, some are even noise. So somewhere we have to have the threshold to ensure that we are considering the very essential ones. Synonyms also include reporting level and reporting limit. I'm sure you might be using these terms interchangeably. And as I was saying, the disregard limit, older term, it still appears in some of the monographs. However, the ICH Q3 term reporting threshold is very much preferred. And also, the peak responses must be corrected by the RRF when the information is provided in the individual monograph, because we know the RRF is so very important in coming to the actual value of the impurity that we are reporting. Then there is also a few glossaries on identified impurity or degradation products, specified impurity or degradation product, whether it's in your specification, then about the total impurities or total degradation products, and unidentified impurity or unidentified degradation product, whether you're in drug substance or you're in drug product, and also unspecified impurity or degradation product. So there are a lot of different terms which I use according to their criteria. There is also a new general chapter, and where we, as I was referring before, it promotes a risk-based approach. And basically here, the threshold-based approach is described in ICH Q3A and B, which used for the reporting, identification, as well as for qualification of those impurities. Higher thresholds may be applied, but it's very much required that the justification is very scientific according to ICH Q3A and Q3B. And of course, it doesn't mean that it has to be always higher thresholds. As we were saying, the justification on scientific ground is very important. So lower thresholds may be required for impurities known or suspected to be unusually toxic, because we know in today's world, as we move forward, there are a lot of impurities that come across. We realize that those are getting mutagenic impurities. So it's always good to do those pre-work over it to understand if there are any chances of the mutagenic impurities, to ensure that the thresholds are lower to avoid those impurities coming up or generating your product. Identification threshold can be established using current applicable regulatory guidances or any other acceptable scientific means. So basically, this chapter contains outline on introduction, identification of impurities in drug substance and drug products, analytical procedures as well, reporting impurities in DSMDP, few acceptance criteria, as well as qualification part. Let's have a look at few ICH guidances as well before we actually go into the center of the area on reporting threshold, where ICH has very well defined those impurities in drug substance with respect to MDDs. I'm sure many of us are aware of this table, how clearly it is defined, whether it's less than or equal to 2 grams or greater than 2 grams, what should be our reporting thresholds, identification thresholds, and qualification thresholds. Same goes with the drug products. Those guidances and guidelines are very much in place with respect to the maximum daily dose. Maximum daily dose is very essential because that's where your reporting threshold, identification threshold, and qualification threshold could be set up. And those range which is given is right from less than 1 mg to greater than 2 grams. Great. So as we were talking about reporting threshold, now having those background completely in place about ICH, FDA's, USP's general chapters on impurities, what different types of impurities are in place, what challenges we have seen, right? That it's not only just one product that we get end of the day, it comes with impurities, right? And to calculate those impurities, reporting threshold plays a very vital role. And USP NF 2021 issue 1, November 1, and which got posted in November 1, and it got official on May 1, covers so many general chapters and which is very much related to impurities. So let's have a check on reporting threshold history, since we are talking about reporting thresholds in particular. Well, as we go before 1995, USP begins including an analytical method parameter like disregard statement, disregard limit, reporting threshold in organic impurities test, describing the level above which impurities should be considered and reported in their total impurities. Then come the era between 2016 to present, where there were concerns started coming up from FDA about the numeric limits for RTs, that it should not be included in the drug product monographs, as the idea was that the RTs should be left as an application assessment issue, and it should not be generalized because different dosage forms could have different reporting thresholds, and it should be based completely on those application assessment issue. August 2019, where the USP published this compendial notice and proposing the removal of RTs in USP NF monographs, considering FDA's comment and FDA's concern in mind. It's very essential to take even stakeholders' reviews before taking any step further. And also in November 2020, we host a first round table as well on the reporting threshold. So in this roundtable, as you've got the challenge that you're facing on this reporting threshold, and we are mentioning those numeric values, the idea from FDA was this numeric value should be avoided, and this reporting threshold should be completely based on application assessment issue. And we started reaching out to stakeholders to take their perception and their suggestion as well before taking any crucial step further. Having this November 2020 roundtable advantages was to have this COVID compatible format, virtual, so many people were present. It was a flexible engagement with the stakeholders, and really a good participation observed from manufacturers, EDQM, trade associations, consulting firms, and even COE advisors. We received really a good amount of comments. FDA also provided informational content, but the challenges were they were unable to interact via the written format. And concern was also expressed by the participant that this posting written comments over an extended timeline may not result in sufficient engagement. So we took everything into account from this roundtable and from the companion notice that was published in August 2019. And what we could see from this roundtable, the three approach was like the approach one that came out was including the reporting threshold, the way it is going as of. So we could see that 73% commenter supports this proposed approach of including reporting thresholds, and 27% opposed it. Approach two, where removing reporting threshold was proposed, and here there was 11 commenters where like 91% opposed it, that we should not remove the reporting threshold, and 9% didn't have any opinion expressed. The approach three, which was a really interesting one, which was a hybrid approach to reporting thresholds, where we could see that 63% supported this approach going hybrid way, and 38% didn't have any opinion on it. So specific comments that we receive, because it's very crucial to even go out beyond and to understand those comments as well. So few comments that we receive were like listed here: a reasonable compromise to address the needs of different users, that is both testers and submitters. Also, there was a comment like products not within the scope of ICH Q3A and Q3B, because we know, right, it doesn't cover everything. There are a few products like fermentation products, semi-synthetic products. So those should be excluded. There was also a comment on the implementation of the approach may be complex and it may subject to interpretation. So users will require those detailed guidance, which is a very valid point, that it's very essential to provide those guidance in detail. So it will be very easy for them to follow it well. The concept that users must verify the suitability of the selected RT in conjunction with the verification of the analytical procedure should be addressed in the applicable general chapter. So all these points were so very valid and on the same line of discussion that we were having. Well, so this newest proposal that we came out with based on those discussions, those comments, was having something in place on user-defined reporting thresholds, where the format is to introduce it at the second roundtable, which held on June 29. So followed by that November roundtable, we had this another roundtable on June 29 that removes those prescribed or prescriptive numeric value for the RT from the monograph, which address FDA's concern. And we replaced it with a link to new general notices containing detailed guidance for a user-determined reporting threshold. So this is just a proposal and the work that is in progress. So that's the format that has been introduced in that roundtable. And the informational note that suggests RT that may be suitable based on presumptive MDD provides a helpful hint. So it's only a good suggestion and the recommendation, and further supporting the USP user as per their comment, so they can go back to it and assess their products very well. And also at the same time, what we kept out of scope was the drug substance and products monograph, which are outside of ICH Q3A and ICH Q3B scope, like biological, biotechnological, peptides. Those will not be considered for this. So this is something as a format, we'll see it as whenever in the future this will come up based on those roundtable and the challenge that came up, and we see the opportunity to work with the stakeholders to take everyone's suggestion before moving forward. And as we see that when we start revising this general notice, something like this will appear. These numbers are just for illustrative purpose, but this is something we can expect in the future with the monographs coming in. So in general notices, the points to be addressed is, as this would be the last slide for reporting threshold, and then we'll move on to excipients part to cover those aspects on the impurities. The few really good points that we could see that need to be addressed in general notices when we start developing it, that this section is applicable only when specified by the monographs. And also, it should provide those guidance on how to determine ICH Q3-based reporting threshold. And manufacturers use the value approved by their applicable regulatory authority wherever they file it. Those regulatory authorities' applicable guidances should be taken care of. How to guidance based on MDT for other users of the standard. This should also be present in this general notices as a guidance. And also to provide tips for the rational selection of RT based on other factors, including acceptance criteria and nature of the impurity. And also at the same time, assign this responsibility to the user because they know their product very well, and so that they can ensure the suitability of the analytical procedure to the level of the user determined reporting threshold. So these are the last two slides that I thought of covering with respect to excipients, because we know that when we talk about impurities, it's not just related to drug substance and drug products, it goes beyond that. So these are the few work that we are working on, even impurities from the excipients front. We recently had this workshop last week as well on it. So there was a similar article we published in PF 44.3, which actually falls calendar-wise in May and June 2018. The proposed idea here was about the view of the USP excipient expert committees on the complexity of the excipient composition and impurities. We tried to define simple excipient, complex excipients, what could be excipient compositions, and what could be excipient impurities. Also, some direction and guidance has been recommended in standard setting processes when it comes to excipients. And what we provided is the few examples, because we know, right, along with the proposal, what helps is few examples. So we provided few examples of challenges faced by USP in setting specifications for different components, and also examples of the current approaches in setting specifications for excipients components with case studies and the decision tree. Also, there was a similar article, USP responses to comments in PF 46.5, which falls under September-October 2020. And this last slide, where we actually, had this survey, and it's like on the guide, the USP excipients expert committees and PUTs on development of documentary standards when it comes to excipient impurities. The specific research objective behind this survey was to collect stakeholder feedback on technical issues they face related to setting specification when it comes to impurities and excipients, and also to better understand current industry practices, because we know, right, it keeps evolving. So it's always good to understand those current industry perspectives and their practices around specific comments, components, and impurities of excipients used in pharmaceutical space. There were 47 respondents qualified for it. They completed the surveys, and results are being analyzed with caution because these numbers compared to survey-wise is pretty low. But USP is planning to launch another follow-up survey to more broadly engage those stakeholders on the topic. So these are on the excipients front, and what we try to cover throughout this presentation about the challenges and the opportunities that we see in case of reporting threshold, which is such a good and very crucial parameter when we talk about organic impurities in the pharmaceutical world. And with that, I would like to thank you all for your attention, and I'll stop here if there are any questions and pass it back to Anurag. Thank you.
Thank you, Amjad. If I may request, if you can stop sharing the screen so then we can display you. Yeah.
Sure. Yeah.
Yeah. Over to you, Anurag.
Yeah, great. Thank you, Amjad. Thank you very much. Deepak, if you want to project the formal certificate of appreciation for Dr. Amjad. Once again, thank you, Amjad, on behalf of the APA and the organizing committee. Thank you for taking your time and helping us close this nice theme with the regulatory perspective. Great. So I think we can get into the panel discussion, and maybe we go. Before that, I'm requesting all the participants, if you have questions, please also mention whom you are wanting to address the question, so that I can directly take that question with the speaker.
So if
let's
I may,
go with.
yes,
go ahead.
so would just quickly request Mohinesh as well as Ravi to, if you can switch on your video, then you can also be projected for the panel discussion. Thank you.
Perfect. Okay. So let me go with few questions which are somewhat linked to what I asked to Ravi, and those are on the drug excipient compatibility protocols, etc. So one of the question is very high temperatures like 60 degrees Celsius, 95% RH, how realistic or useful they are from extrapolation perspective. Another linked question is whether surface area is important. I think you touched upon that while responding to or during your presentation, but still for clarity, if you can just add some more light on to how surface area is relevant or important in the conducting an outcome of these drug excipient compatibility studies.
Yeah, so thanks for asking that question. So first, let's understand why we do a drug excipient compatibility studies. So the objective is very clear that whenever we make a formulation at a later stage, there should not be surprise of having a non-degradation product or the stability is not fulfilled. So we want to do a drug excipient compatibility studies before in hand. Now, the second question comes during how much duration we would like to complete a drug excipient compatibility studies, because we cannot keep on stability for every trial. If we put every trial on stability, there is no meaning, right? So we want to complete the understanding or a risk assessment as early as feasible. Now, what we are trying to do is we are trying to make a condition in such a way that it is so much accelerated that we get a results early enough. And that is the overall objective of a drug excipient compatibility studies. Now, the question is very valid that should we put a 60 degree or 80 degree or a more RH, and should we generate a conditions which formulations may not achieve in any moment of time? And that is very true. So generally what happens, a stress condition has given which is higher than the accelerated condition with the objective that the degradation products or a drug excipient interaction products formed early enough. Now, as Amjad was talking about a reporting identification or a qualification threshold, we are looking in actual formulations, any impurity above 0.05 or any impurity is crossing the identification threshold of say 0.10% and so on. But whenever we do a drug excipient compatibility studies, generally we look for the impurities which are more than 0.5%. So we always try to understand, okay, what are the pros and cons, and it is actually a balance with the time or a durations which we can decrease and how we interpret the data. So your question is very valid, and I was trying to convey in my presentations, there is a need for having a realistic stress conditions to avoid a false positive and false negative by particle size reductions, effective surface contact, and so on. So that is there. The second question was obviously the surface area. The surface area is directly proportional to the interaction. If two particles are not in contact, obviously they are not going to interact. So they have to come in contact, and how much they come in contact is effective surface area which are shared by both the particles. So the idea is to increase the surface area. If you increase the surface area, obviously the interaction will be much higher, and as a result you can decrease the durations and decrease the stress conditions and so on.
Perfect. Thank you for clarifying, Ravi. Let me go to another question, which is for Amjad. So the question is, agency expectation is to characterize the impurities, right? We all know that. And often these impurities are below the identification threshold. So how critical it is, and what are your thoughts in terms of whether any characterization work should be done for such impurities which are below the identification threshold?
Thanks, Anurag, for that question. So yeah, identification threshold, and if we see those limits, like, if it's below identification threshold, I would say you are still in a good zone. But if it goes above identification threshold, it becomes very essential to start identifying it and to understand if it goes beyond qualification threshold in your studies. It's very essential to start qualifying it as well. But nevertheless, as we always trying to understand in today's world, like we've seen, right, from the cases like nitrosamines, it just came up all of a sudden from so many areas. So that's the whole idea behind it, that for the manufacturers to keep that in mind whenever in their impurities, if they expect something coming up, if it's not even going along with those guidances and guidelines which are in place, but if they see there is a chance of this could be a mutagenic impurities, it becomes more sense to go deep down, evaluate, and don't just restrict yourself to those limits provided in guidances or guidelines. But it's always a really good proactive work that the manufacturer could do or the user could do to evaluate it on all possible ways, whether it could be a mutagenic impurity or it is not. And that's why what I was emphasizing, and I couldn't stress enough on that point during my own presentation, was that it's very much required for manufacturers, because product is their baby, right? So how best they understand that, and no one else could understand that better. Now, when we see in the case of nitrosamine, the limits are much lower, right? And that too, we know these are all interim limits. The idea is to not have them in place, but this is just to avoid those shortage of products. We have some interim limits in place from the guidelines. So the preference and the suggestion would be just try to ensure that this couldn't be a mutagenic impurities or something which will be harmful to the patients. If that is the case, or there is any even a chance, don't rely only on those thresholds. Go ahead, understand that impurity very well, and then accordingly set those specifications or control it.
Yeah, that's right. Thank you, Amjad. In fact, I think to summarize, the bottom line is that it's a question of being proactive. It's a question of being understanding what you're seeing and what is happening in the product. It is also a question of risk mitigating of future potential failure, and things do change. We all know about various impurities which initially were not considered, of concern, but later on they really became a matter of concern when the manufacturers had to even reformulate or withdraw the products, or meet the tighter limits which were established later on. So being proactive in doing that is really essential. Thank you. So let me go to the other speaker, and this is general question. It's from my side. How do you extrapolate or leverage the understanding of extractable leachables area in the parenterals and in the parenteral products to the other non-parenteral dosage forms? And for example, liquids which are for topical or oral administration, the semi-solids or topical products which are packaged in various kind of polymeric and plastic containers and closures. So anything that you would advise to the formulators, to the formulation scientists and manufacturers, Monish?
Yeah, sure, Anurag. Very good question. So first of all, we can see that there is a guidance from FDA. It is 1999 FDA guidance, which talks about the route of administration and likelihood of interaction. Based on this topic, they have categorized different container closure system and different formulations on the basis of whether the risk is high, highest, or low. So if we see that guidance, so keeping on the highest side, it is inhalation solutions or YNDPs, and on the lower side, it is solid orals. So now coming to your question, how to extrapolate the data. So if we have a extractable data of a particular container closure system, suppose a vial or any container, and if in that container we are going to keep parenteral, or we are going to keep some other liquid formulation, syrups or something like that, In that case, we need to see that what is actually the formulation of that product which we are going to store in that solution, in that container. And based on that, we can extrapolate and see that based on my current pH of solution, my current components of my formulation, what are the extractables that can reach into my product. So that can be done based on like what is the, if it is an aqueous formulation, so definitely nothing is going to come which is highly non-polar. So that is the way out where we have to see that based on the formulation and the components of that and the pH of that, how and what extractables can be our potential agents. So that is the way of extrapolation. So it is all not only for parental, because I can see a question over there where it's written whether it is only for injectables. So as I already told, no, it is not only for injectables, it is for all the products. There are cases when the tablets are being recalled due to the presence of some misdiorder due to tribromoaniso. So currently the regulatory agencies are focusing with every data, there should be a E&L assessment as an integral part.
Maybe in continuation and follow up, if you can also tell what's the importance of having the likely or proposed commercial product used in these kind of studies, because many times the ink or the components of the label are important part of the package, which can result into different extractable leachable profiles. So any thoughts around that?
Yeah, definitely. So generally the CCS is categorized into primary containers, secondary container, and tertiary container. And along with that, there are studies like ink migration study, label studies. So there are these are all part of our leachable study which we perform, and a part of extractable or ink migration study, where what we do that first we focus on the primary container, that what actually can come from my primary contact material, because that is the most worst case, because that will be the most duration when my primary container is in contact. Then we also check what is the possibility of getting ink migrated from a label or any other markings into my product. So that is a part of the leachable study, and it has to be performed along with the complete transaction.
Great. Okay. Let's go to drug excipient compatibility studies one more time. And Ravi, there are still some questions I think are left from previously posted questions. So how would you include water for about 95% RH in proposed protocol? I think this is something more from a practical perspective. How would you create such an environment where you have about or more than 95% RH in a closed study?
Yeah, so a very good question. So what we are trying to do is, when we were trying to put a stress conditions, try to increase the RH. And for making a RH, a conventional way is taking a desiccator and put a saturated solutions of different salt and try to make it to a different RH. But there are ways, say for an example, at a 60 degrees C, if it is a closed conditions and the water is available, a water will get evaporated and make the environment saturated at a particular temperature. That is called a saturated relative humidity. And a saturated relative humidity at a different temperature are set. So what we do is, if you see on the slide, what we have done is we took a GC vial. In a GC vial, we put an insert, and there is a glass insert which can fix to a wall of the GC vial, or you can even put down, which is filled with water. Now you crimp that, so it is a closed environment. And now you put at a 60-degree temperature, basically water will get evaporated. And a water will get evaporated till it reaches to a saturation relative humidity. And at a 60 degrees C, a saturated relative humidity is 95. So what you need to do is, you need to just close the container and put it at 60 degrees C. That's all. You need not to measure the RH. You need not to do anything extra for getting that RH attained within the system. So it's much simpler In terms of a practical aspect, you need to have a closed systems, and what we have used is a GC valve.
Great. So I hope that answers that question. And let's move from the protocol design of study to a slightly different line for you, Ravi. There is another question, which is to expedite the studies, are there possibilities to use latest tools and system available like ASAP, Accelerated Stability Assessment Program? So what are various options? Probably that's part of the question. And another linked question is, you'll get more flexibility for worst conditions like 0 to 100% humidity, 40 to 90-degree temperature to expedite research. So any thought the value of such tools? So two parts of the question. What are various options available or tools available, and what is the value of such tools?
Before you answer, a quick request, if at all possible for you, if you can switch on your video, so the spotlight can be on you. Yeah, thank you.
So, sorry, Deepak. So the first question about ASAP. So ASAP is mainly used for prediction of stability. If you see on the mean kinetic equilibrium and the modified Arrhenius equation, what generally happens is you put the formulation in certain conditions and try to identify a rate kinetics. And for any kinetics, it is essential that it is very much essential that you need to have at least 5 data points. And if you have a 5 data points, then you can make a curve and can identify a rate kinetics. In drug excipient compatibility studies, is like a quick and easy or a fast. So there is no point of, according to me at least, it's no point to generate the data at least 5 data points at a different temperature and different humidity and do a self-life prediction. ASAP is more of a self-life predictions. There it makes perfect sense that you actually do the linearity curve or a reaction kinetics, maybe a pseudo first order, or in worst case scenario second order. But in this case, that is not the case. There are other ways to speed up the thing. So say for an example, in our lab, what we do is we put the drug and excipient together and put on a TGA instrument, thermogravimetric analysis instrument. Okay, so what we do is, say for an example, I'm using a HPMC as an excipient, and HPMC has a TG around say 170 degrees C, and the drug is in crystalline. It has a melting point maybe 200 degrees C or so. So, and I would like to just say that, is there any possibility of interactions? I mix the HPMC and API, go till 180 degrees C, kept at 180 degrees C for only 2 minutes, bring it back and take the sample out and do HPLC. So there are multiple smart way of assessing the incompatibility. So in terms of tools, people try to make an instrument, and I think we are also trying to make an instrument which can generate the samples and can do a drug excipient compatibility studies in a week time or in a 2 week time. So that we are also trying to do, but no standardized a guideline or tools available, and there is a huge scope, and that may vary from company to company how they would like to take it further. But there are a lot of scope. I think there was a second question about, sorry, Anurag, could you please remind me the second question?
Yeah, so what are the tools and what are the applications, or how can you utilize those for predictions?
Yeah, so
you covered in a way that you covered.
Yeah, that's true. So generally there is, you can do minor kinetics, but the prediction perspective, if you do not have a choices, on only those cases you do a prediction, and that is feasible. And you can use a DOE also, design experts type of software also, where you can identify a response factor with the 2 data points or 3 data points. And that is very much feasible. We also try to do a similar with the principal component analysis and others.
But another related question from my side is, how can for example, artificial intelligence can play a role in making such predictions with good understanding and model for interaction of drug. Ultimately, all excipients drugs, they are one or the other type of chemicals. So their functionalities, their functional interactions at the molecular level, and how the available data can be utilized for predicting what kind of interactions can happen in a group of, which is, I wouldn't call something very something that we discovered just yesterday, because we've been using, like, so we know peroxide containing excipients, and peroxide can cause some kind of, so, but having said, still is the evolving landscape of artificial intelligence
Yeah,
is
so Anurag, so Anurag, there are a couple of softwares available. Previously, there was a software called D3. Now it is like almost discontinued, and there is a software called Zenith. So Zenith software can do certain predictions, and they may ask for some input data, which may be of a LCMS data. We are currently working at NIPER with Schrodinger software also, and try to understand certain functionality and how it can do a predictions. This is like only based on a risk assessment where the choices are multiple, and if certain functional group is not present in the excipient, you can just ignore. And so there are certain ways not widely used because of one or another reason, but Zenith software is really good.
perfect. So I think there are two, if I summarize your answer, there are two different approaches of handling the predictability and predictions in this area. One, which is chemistry and molecular chemistry based approach, where you predict based on the functional groups and functionalities of the molecules. And the other one is based on the real time data that you generate, either in exaggerated conditions or some kind of model where you are able to generate some data and predict that this kind of interaction can indeed happen, and then that's another way to. So I hope that addresses actually a bunch of questions which were posted. And let me go to now next question for Amjad. So Amjad, is there any specific limit for nitrosamine impurities more than one present in drug substance? I think this is a hot topic for quite some time. Do you want to present what is USP's take on this topic?
Sure, Anurag. And as you rightly said, it's a hot topic. Nitrosamines is therefore, I think it started back in June 2018, if I'm recollecting my memory right. So that's where it all started. And of course, there are a few limits, especially for drug products, because it goes to the end user, right? That's where it becomes very much of concern. And there are lots of, one of the guidance from USP I could share is on General Chapter 1469, which really elaborates very well on the aspects of nitrosamines. And I would really suggest to go over that general chapter, which will help you in understanding nitrosamines aspect from the drug substance, from the drug product point of view. So, yeah.
Okay. And another question, Amjad, now this is from my side, the alignment and harmonization, so to say, between, for example, USP and the FDA is pretty well. I think they're doing very good in terms of keeping up pace with each other. But at the same time, it looks like it's always work in progress when it comes to alignment with the rest of the regulatory world and rest of the regulations of ICH, for example. So any thoughts how USP or what initiatives are happening at the USP level or FDA level to bring more alignment with rest of the regulatory environments and expectations?
Yeah, thanks for that question, Anurag. I think it's somewhere we are trying to strengthen those relationships which is already in place, but we, of course, we try to strengthen it as much as possible. Both FDA, USP are working really closely on how best we can harmonize on those fronts, and of course ICH as well, because we know, right, that if there is any upgradations in ICH guidances, same comes for approvals where FDA comes in, and again, then it follows back on compendias where USP comes in. So we all have different platforms where we connect with FDA on a regular basis to ensure on all these aspects we can cover upon, whether it's on impurities, whether it's on reporting threshold, even on decimal values, I would say, on reporting those impurities. Because we know, right, somewhere before 1995 or 2000, even the way we used to report impurities then and the way we report now has been upgraded from ICH point of view as well. So those are also few areas where we are trying to catch up on with FDA. So same ways, like there are many aspects where we continuously connect with FDA, and FDA shares their concerns, their challenges, and along with the few proposals like we talked about reporting thresholds right now. And USP always try, along with FDA, both coming together to find the best way out, which will work, of course, the safety of patients as a top priority. At the same time, to ensure that manufacturers and all other stakeholders are in alignment. So nothing comes as a surprise to them. So that can fall back, fall them into out of compliance. So that all been taken care of. That's the reason that, we always do those stakeholder engagements. We do those roundtables. We do these workshops at the regulatory bodies. So that's the whole idea behind it, to ensure that this relationship gets strengthened. And at the same time, manufacturers and the patient safety, which has come on the top priority, shouldn't be left behind.
Okay. Okay. Thanks, Amjad. Since we are talking of these regulatory and the alignments, etc., so Monish, one question for you, and that's more for the the extractable leachable still continues, in my personal opinion, continues to be an area where not many companies are very well up to date in terms of what to do. When you do an extractable leachable study, you get a multi-page, several rows, several items of X, Y, Z, and so many number of potential extractable and leachables, and then you have to filter out which one you can be of which one of those can be of concern. So any quick guidance for those who are actually doing this work day in, day out, how to filter, what to look for when you have such a large list? What should be the next step immediately after you conduct a study, conclude it, and you have a multi-page report of that with several hundreds of tens of new or unknown or very trace amount of extractable leachables reported, unknowns?
Right. Really, it means a very good question and a main challenge for all E&L chemists. So the thing is, the first thing which I'll do once I get the extractable data is that I'll sort out the substances which are above my AET. The substances or the extractables which are below AET are not my concern. First, I'll sort out the list that what are the compounds I'm getting above my analytical evaluation threshold. Once that list is sorted out, then I'll take the help of a toxicologist. I'll go to a toxicologist and I'll tell them that this is the list for which they have to perform a tox assessment. So once the tox assessment is performed, I'll get my margin of safety. That for a particular compound, what is the margin of safety for that compound and how it is dangerous for a human being. Means what if margin of safety is high, I don't have to worry about that compound. So based on the margin of safety, if it is lower than 1, I'll take those compounds as my target leachables and I'll perform my leachable analysis. So that is a quick pathway which we can take for proceeding with extractable leachable studies. And
I think the prerequisite for that is that whatever is above the AED, you must identify that first.
Right. If it is unknown, we have to identify it. But definitely, as I told in my presentation, because of these low values, like in PPDB and PPT, sometimes it is not possible to identify them. So in that case, biocompatibility data, the AIMS test, those can be applied to show that that extract is not dangerous for patient.
But then often isolating such unknowns is also not very practical or feasible approach.
Right.
Do you have any suggestion in those situations?
Right. So in that case, if because isolation is not a choice only, because we can't use PEP and NMR for that. So in that case, what we use is generally triple quad MS, Orbitraps, HRMS, which are much more having the libraries and the decimal point, the resolution which they have are much higher to identify those compounds. But even after that, if it is not identifiable, we have to go with the rest. We have to select some surrogate compound based on that tox assessment. We have to justify those compounds, because at least from this HRMS data, we can get to know that what are the functional groups, what could be the probable structure of that, even if it is not confirmed, what could be the tentative idea. So based on that, we can select a surrogate if reference is not available, and we can perform the tox assessment. And also, yeah.
Sorry, go ahead, finish it.
I just want to add something on microsomy, the question which was taken before, so about the limits. Yeah, so about the limits, there is a FDA guideline and a CHMP guideline which talks about two limits, that is 26.5 nanogram per day and 96 nanogram per day for a single impurity like NDMA or NDE. And there is a EU guideline which talks that if there is a multiple nitrosomies, then we should be restricted to the TTC of 18 nanogram per day.
Perfect. Okay, let's move on. So Deepak, assuming that we have a few more minutes to cover a few interesting questions.
Yes,
and
yes,
of course,
please
the speakers have some time to stay for some more time.
Sure, Nada.
Okay, so it's an interesting question, although not directly for compatibility of drug with the excipient, that topic, but certainly related to excipient. So Ravi, I'm directing it to you. The question is that in preclinical discovery, often you encounter, excipients situations where excipients have toxic liabilities for the animals. In the animal which you are using as a model, or where you are trying to study something in vivo, right? For example, using of DMA is toxic to rodents. Now your formulation may have DMA, and although it is compatible with the drug, but I think the question is focused more about the use of such ingredients which might be toxic to the animals in the preclinical research. So share your thoughts on excipients that tend to contribute for toxicity and how to avoid the same.
Yeah. So
open the question for anyone who would want to respond.
Sure. So first of all, I mean, say in a drug excipient compatibility studies, you certainly should do or consider that vehicle as one of the excipient. It can be a DMA, it can be NMP. In very few cases, a certain amount of DMSO that may be there. But the objective in the rodent or animal studies, either it will be to understand pharmacokinetics or getting certain markers, or IND tox studies. So the objective for those excipient is to deliver or understand a toxicity or a pharmacokinetic associated with the compound. So before we move on, at least in the case of preclinical studies, whatever tox formulations we are making, we generally want to have at least a very preliminary drug excipient compatibility studies, not extensive. And most of the companies, maybe Pfizer, BMS, Novartis, or any of the company, they have a standard platform type of formulations available, either a drug in a solution or in worst case suspension, and in a worst case scenario, any oral other than liquid formulations. But the objective at a preclinical stage is to just ensure that there is nothing else formed from a drug structure. It's no degradation product has been formed. And the objective there for a drug excipient compatibility study is very preliminary. It means, in fact, people do not give a name of a drug excipient compatibility. They just do a stability on the platform formulation. Now, coming to the regulatory limitations and other things, when it is more challenging when in phase 1 it is like a drug in bottle or drug in capsule type of formulations, and the salt get changed. If the polymorph get changed, a drug excipient compatibility studies may not require to be done again from a regulatory perspective. Regulatory will not ask to give a drug excipient compatibility studies when there is only a polymorph get changed. The other thing is same. But if the salt get changed, or if it is converting to a crystal, or it is a co-solvate or a solvate was formed, in those cases regulatory can ask for again a drug excipient compatibility studies, because your component got changed. And at least in few of the companies, what people do is people do a drug excipient compatibility studies with putting 100 microliter of water and so on. So at least in the cases of a hydrate, people try to justify that we have not done the drug excipient compatibility studies, because in our drug excipient compatibility studies, water was part of our studies. And in case of a hydrate also, the chemical entity is same. But that again depend on company to company, how or how much risk a company would like to take. So that all depends on case to case studies and how you justify it, basically.
Yeah.
I can only just add, since you really raised a good question, Anurag, on front of animal health, right? When we do the studies, we talk about patient safety all the time, but also what good to see this days coming up is the awareness about animal health as well, because there are few even testings where just to determine those assays, animals are used to get it done, and these are all long back methods which are in place. I think few of in radiopharmaceutical drugs, you would see this is there in there. So the good thing to see is that there is an awareness, and users and manufacturers, compendiums are looking forward to move away from such tests and bring something where this animal would not be impacted and it could be directly done. So I think it's good to see overall on the animal front how this awareness are coming in. So yeah, just those are my 2 cents on that.
Yeah. And I'm not sure if the person was really referring to animal health drug development or a human health, but I think the common reference was to preclinical stage of the drug discovery. And in my opinion, if you know that a particular excipient has a safety or a tox implication in the animal model where you are studying, irrespective whether you are doing the study for efficacy or safety or tox, try to avoid it, because otherwise you'll have confounding results, and you'll not make out what effect is because of what. Whether your drug is acting or whether your excipient is creating something which you don't want to see or you want to see. So irrespective, I think try to avoid those situations. And if it is unavoidable, then have a way of discriminating what is coming from a known tox effect on the animal. What is your intent of finding by administering such a formulation in the animal?
Indeed. I think that discrimination will definitely help, because we know, right? Sometimes instead of leading you in the right direction, it misleads you because it was excipient instead of that active drug. Yeah, very true.
Yeah. So there's one more question, Amjad, for you. So can you throw some light on how the studies are conducted in industries and related regulatory environments? Requirements, sorry, not environments, regulatory requirements.
Sure, Anurag. So I think basically I could see there are 2 parts to this question. One is how it is done with respect to related substances, and another is the guidances which are in place. So to take the first question first, I think there are many ways where you can have a foolproof testing on these related substances, ensuring like, considering even literature survey as well, I would say, and existing those developing methods for those APIs, keeping in mind those route of synthesis that comes from, because with the change in process, you can see the change in the impurity profile as well, which plays a very important role to have that knowledge in place, that what process has been followed. Accordingly, you can develop your methods, ensuring that it's very much stability indicating, and doing a lot of other studies as well, rather than just doing those testing and developing your trendings. Generate those stability data over the period of time, along with force degradation, and keeping even Arrhenius equation in place. That helps at times, and as we heard about, right? That also helps a lot in like forecasting that how your drug would behave. Along with that, I would say after doing those, there are so many databases in silico tools are available these days that we can take advantage of to ensure that you are taken care of all the possible aspects, that how if this could be going to be a very good scenario. For example, in nitrosamines, when we talk about ensure that how if there is any presence of secondary amines in your API, and if there is any nitrates or nitrides and excipients, how this combination, there are possibilities leading to formation of nitrosamines in your product. Do those theoretical study as well. So there are so many different aspects that you can take care to ensure that your related substance of API that you are studying and developing and establishing those data are foolproof, and you're not seeing any surprise in the future. And coming to the second question on regulatory guidances, I think there are many guidances. Even I speak from ICH front of view, there is Q3A is on drug substances related impurities, Q3B is on drug products, C is on residual solvents, D is on elemental impurities. And the recent one which is coming up, which I am looking forward to, being very close to my heart, the topic on extractables and leachables. So it's really good to see even those chapters are coming in Q3 from ICH. And even if you refer to USP, there are chapters like 1086, which talks about organic impurities, 476, which talks about control of organic impurities, 467 is on residual solvents. And also I would recommend to touch upon 232, 233 for elemental impurities. I think there are so many references you can touch upon that will really give you so many guidances and references to ensure that you have a really good package of related substance with you.
Wonderful. Yeah, thanks, Amjad. So let's go to Monish. There is a question for you. So which is the best solvent for aqueous drug product to do the extraction or extractability studies?
All right. So a very, very tricky question, I'll say, because to demonstrate or to say that which is the best solvent, it is actually impossible. It totally depends upon your formulation. But considering it as an aqueous solution, I'm considering it will be consisting of some salt and water. I'll prefer to go with aqueous buffer solution like pH 2.5 aqueous buffer solution and pH 10 aqueous buffer solution. So goes towards acidic and basic side with the buffer solution for the aqueous drug product. And if you want to do a little more to be more confident, I can also include one solvent like the water is to IPA 50 is to 50 to demonstrate the worst case. But definitely I'm not going to go with hexane or pure IPA for the extraction.
Well, I think that probably covers generally general questions. There may be one or two. So let me quickly go to there's one question which I believe partly we might have addressed, but still for the sake of completing it. Can you comment on drug excipient compatibility of API with individual excipient or with their grouping? Okay. So maybe the participant wants to understand if you can actually group the excipients or the APIs together by their functional group and draw any kind of drug excipient compatibility conclusions by just doing with one of the representative material of that functional category or class. I think the chemistry oriented functionality probably is the question.
Yeah, let me try to answer this question. And the question is very valid when we can only do with the drug and excipient that is called a binary mixtures, and people do with the binary mixtures, and people have done with the ternary mixtures also. And then the concept came about n+1 and n-1 type of mixtures. So let me try to give generally what is n+1. N+1 means you take the API and the most common excipient, which is like a diluent, for an example. So in the one experiment, you make API plus excipient 1. The another experiment, you can do API plus excipient 1 plus excipient 2. And so on, you can increase the number of excipient. That is called n+1 study. And there is a study called n-1 study. In that, in the total formulations, you omit one excipient by one. What it means is, say API plus 7 excipient. So the first study will be API plus all 7 excipient. Then it will be API plus 6 excipient, and you omit one excipient, say excipient A. In the next study, it will be again API plus 6 excipient, but instead of omitting excipient A, you are omitting excipient B, and so on. And there are a lot of DOA designs also coming where, from a chemistry perspective, you put certain excipients as in a group, and that is also feasible. So they are in a DOA, the categorical designs with the numerical, and combination of a categorical and numerical is feasible. If somebody is particularly interested, please drop me a mail and I will try to help you guys.
Great. I think that we are left with a couple of questions which are really very generic, and I was reserving those for the end because I think all of you can comment on those. So let's say I'll go one by one for those two questions, and you can put your thoughts. So one is, although it is addressed to Ravi for you, but I think there can be different perspectives, and the question is really generic. What all the challenges and regulatory limitations when you are transferring preclinical formulation to clinical study? So I think that's very general question. So that's why I thought, okay, you can just put your 1 minute thought or whatever comes spontaneously to your mind from your own perspective and lines of work that you do. So any challenges that you would want to highlight when a preclinical formulation is moved into the clinical?
Yeah, so for me, preclinical to clinical move, if you have done a really good force degradation studies only with the API, those data only goes from preclinical to clinical, because in 90% cases, whatever excipient you use in preclinical, you are not going to use in clinical, because clinical setup, it will be like definitely a significant change will be there in the excipient. So from drug excipient perspective, if means whatever I have done so far, and what I recommend also is take only a force degradation data forward, nothing else on from drug excipient compatibility perspective. In a different clinical phase, certainly you need to take certain data and keep in mind from phase 1 to phase 2A, or phase 2A to 2B, and then till phase 3. But when you are transferring a phase 1 to phase 2A or 2B, you should be very mindful about the particle size of the API, as well as the form of or polymorph of the API, because that may certain time give a sudden surprise that, okay, this was not happening, how come this started coming? So you should be mindful for that. I think a lot of people are there who can talk more on the regulatory aspect. I'm sure Anurag, Amjad might have worked significantly from preclinical to clinical, and they can talk on regulatory aspect. There are multiple things will be required from regulatory perspective.
I think there could be a lot of challenges that you could face when you move from preclinical to clinical, because there are very few commonalities that you take forward, because preclinical is a very initial step that you take. But as you move towards then progress in your clinical phases, 1, 2, 3, there are so many changes comes in from every angle, right? Whether it's from the process, from the routes, from the your excipients that you're using. And accordingly, you could see those changing in the trends as well. And when you start comparing those with the initial or the previous one, you cannot, because it's not apple to apple, right? So those are a few challenges which you will definitely encounter and which are the major one. And of course, there are a lot of them, but those are the initial steps. And as you move forward, the idea is to ensure that whatever issues or the challenges is coming across, you are taking it one by one, you are addressing it, and you are moving forward. That helps a lot. Many of the users take that approach, pursue that approach to ensure that they are not getting those issues as they progress in their clinical trials.
Anything from your side, Manish?
No, I'm totally aligned with Ravi and Amjad that definitely there are very less commonalities which are going to be there from pre-formulation or preclinical to clinical stage. And definitely the only thing which we will be referring is the degradation pathway of the API to see that what could lead good as a metabolites or something during our clinical phase.
Yeah, and maybe I would add my 2 cents here that when you're moving from preclinical to clinical, you're bound to enter in some kind of changes happening, right? For any reason, let's say pharmaceutical development, manufacturability. For one or the other reason, you might start tweaking the formulation, start tweaking the drug product, and one has to be mindful of what quality attributes you were having in the product earlier versus how those are changing as you keep making progress into the clinical phases, various phases. And you may have to always keep in mind that whatever has given you good product initially, that's what your ultimate objective is. And if something is changing which can have an impact on that, then you have to be mindful and proactively think whether to do something or not do. And also be mindful that when you are progressing into the the preclinical and clinical formulations, as you go into the more and more advanced stages, there is less and less of expectation and flexibility or freedom to make changes. So anything that changes then has to be also bridged because agency would not otherwise deem the formulations to be equivalent if there were some differences in various stages of the development. So that's my perspective anyways. I hope that was good insight for the person who asked this question, though the question was very general. Let me go to another very general question. And again, this doesn't probably relate to any of the presentations today, but still maybe worth having it discussed since we have experienced people present in the seminar today. So the question is, most of the times in marketed drugs, the tablets get melted inside the packaging. I slightly beg to differ on this, the usage of the term most of the times in particular. But anyways, there could be some instances, agreed. In this case, what can be concluded? Now, that's a very, again, general question. I don't know who wants to take the first stab and wants to answer that question. I can put in my thoughts as well. And with that, we can then.
Right. So I'll take up this first initiating this. So definitely means I agree. Means using the term most of the time is not suitable over here. So yes, but if there is a barrier deficiency in the container closure system which has been used to wrap that tablets or to store those tablets, there's a defect in those containers, then this type of situation can arise. And that generally happens where with some local companies or something who are not branded enough to maintain that integrity. So in that case, what can be concluded? The only thing which I concluded is don't use those tablets and don't want to go with that brand anymore. That's it.
Yeah, what I could add over here is, of course, this when we see those tablets getting melted inside the packing, I think one of the things that could come in mind is the about the stability studies, how it has been done on those different temperatures and humidities, whether all those 4 climatic zones has been considered very well, and also about their supplies, where these products will going to be supplied or marketed. If those climatic zones has been considered, even the transportation part, right, even storage part, because it's not only about manufacturing and supplying it, But what is very essential to be considered to avoid such instances is the storage and the distribution and those climatic zones. If those has not been considered, it can definitely lead to something like this. And when we heard about tablets, even in fact, if I look from the lens through extractables and leachables, I think these are the packaging. In fact, those are the low risk dosage forms, tablets, right? It comes at the most in the blisters or those areas. It becomes very risky when we talk about high risk dosage forms from the inhalation point of view. But in tablets, it shouldn't be the case. So I think those regular basic guidances could be followed from the climatic zones point of view, stability studies. I think that should take most of the care of it to avoid such instances.
Yeah, from my side, from my side, there should not be no such instances. It like directly failed in a appearance, and that's all. I mean, so from product and development perspective, as R&D, somebody should have thought about the TG of a polymer, what has been used, or why it is coming on a melted state or a glassy state for chipping and other things. But in no such instances, the specification should pass that. And if you are from from analytical side, being a quality gatekeeper, straight away fail the batch.
Yeah, and I'll probably be more direct in saying that if something like that happens in a commercial product, that means something has been missed in the development stage big time. I think, as Amjad was saying, that there are standard protocols or standard study designs which must be done during the development. Various climatic zones. Think about what is the risk of the formulation or the product behaving, or something like that can happen for the product when it is in the real life situation. The finished dosage form in commercial pack, which is exposed to the conditions, transportation, handling, etc., when it sees the reality of the world. So anyways, bottom line, I think it shouldn't happen. In summary, it shouldn't have happened. If it has happened, do go back to the bench and investigate why it would have happened and try to avoid those situations for future. That can be the only advice. Well, I think that covers all our questions for that matter. And I didn't see anything added more.
I completely agree, Anurag. And,
okay, so there is...
Anurag, while you are taking, are you planning to take one more question, or we are done with the questions?
Well, no, I think one of the other participants probably just added a comment and opinion. Okay. So I'll just read it out.
Please go ahead.
Yeah, it's market complaint and should be reported appropriately such cases to avoid the severe impact and putting patient on risk. Yeah, very well said. Those kind of cases, by any chance, if anybody sees, then immediately it should be reported back. Pharmacovigilance is the way to report it to the company or to some agency and make sure that the information channelizes so that the risk is avoided for continuation of such a product being administered to the patients.
Thank you, Anurag. And while I request you for your concluding remark, I think would like to congratulate all the speakers and would like to thank them for such an interesting talk, followed by very, very interesting discussion, interaction, what we had. And I think as we have seen, these are CMC is one of the very important area. And that's how you've seen a lot of questions also coming in from all the topics what we have talked about today. So while Anurag, you would be presenting the concluding remarks, I would just launch this poll. So all the participants and even panelists who would like to participate can just respond and choose your responses on the poll and submit that. So within 2 minutes, then even the concluding remark ends, then we will continue with our next proceeding. So let me just launch this and then would request, so it is launched on your screen. You would see just 4-5 questions about today's webinar. So would request everyone to please submit your responses. And now I would request Anurag for his concluding remarks for today's webinar, and then officially we can end this. Yeah, over to you, Anurag.
Thank you, Deepak. Well, formally, I must thank all the speakers, starting with Suri and then Ravi, Amjad, Monish. Thank you for taking time. Appreciate it, and especially Suri, as he was dialing in at almost midnight for him in US. And also that all the other speakers who have taken time out of their Saturday mornings and their personal times to make presentations and give very good insights on the topics that you touched. And the interest of audience is very evident from the kind of questions, the number of questions and quality of questions that we had. And in my opinion, it was a very good discussion at the end during the panel discussion, very relevant questions asked, and I'm sure the answers could satisfy and provide the right answers to the right details that people were trying to understand. With that, I would like to thank everyone, all the participants as well for taking time and attending the seminar today. With that, I'll give it back to Deepak to conclude.
Yes, so very quickly would like to mention that it's my mistake. By mistake, we have launched the wrong poll. So now on your screen, you would see webinar 12 poll, where all the 4 talks are included as options. So now you can submit your responses. So I'll keep this screen on. The poll would be live for another minute or so. So you can submit your responses. Because in the earlier poll, I think there were only 2 talks mentioned, but in this poll, all the 4 talks are mentioned. So sorry about that, yeah. And just would like to re-emphasize what Anurag has just mentioned, a great session. So thank you to each of the speakers, starting from Raj, Ravi, Mohinish, and Amjad, great session. And thank you for your valuable time and insight into the subject and clarifying all the doubts. In case if further queries comes up on the Boston DB21 email, then I will get back to you. And would like to also thank Anurag for doing a great job as a moderator and all the audience for their very, very active participation. So thank you all, and would request everyone to again block your calendar for next Saturday, because we don't have a webinar on next Saturday, 2nd November. But 2nd November is our poster session day. So all the shortlisted poster presenters, they will get to present their poster live in the poster session, which will happen on 2nd of October.
Yeah, second, I wanted to correct that. I think Deepak.
Yes.
2nd of November. So.
Oh, okay, yeah. So sorry about that. So it's 2nd of October, and yes. Would request again all of you to please block your calendar, and let's encourage those poster presenters by participating in that. So I know 8 minutes is too less a time for a presentation, but I think at least to get an exposure to present in front of an August audience would be a great encouraging thing for the students and the researchers who have submitted